Oh, so good evening everyone guys. Yeah, hello. I think everyone of you are able to listen to me and see me. Chalo, badhiya. So let's get started right now. And Divya has got one doubt here right now. Divya, whatever question you're asking, the pyruvate complete oxidized, that is a question from FM. Like that. Okay. And for the complete metabolism of glucose, right from the very beginning, it will be 32 ATPs. You can calculate. iPad says, sir, please tell me about the BCKD enzyme. BCKD is branch chain keto acid dehydrogenase enzyme, and that is going to metabolize branch chain amino acids, valine, leucine, isoleucine. Branch chain keto acid dehydrogenase is going to metabolize branch chain amino acid, valine, leucine, and isoleucine. And deficiency of that enzyme will lead to a disease, and that is known as maple syrup urine disease. ठीक है. I told yesterday during the class iPad C how did keto acid came in that one. When from the amino acid, when you remove the amine group, the remaining part will be keto acid. After removing the amine group deamination, the remaining part will be keto acid. Chalo. So now today let's get started with our today's class right now, guys. So please write in the heading as nucleic acids right now, sir. So today it's all about dealing with DNA, RNA, and all those things, and that will finish off biochemistry today, sir. And that will finish off our session today. Okay. Now welcome to the nucleic acids. Remember, the nucleic acids are of two categories inside of a body. In human beings, the nucleic acids are of two types, sir. One will be your DNA, and another one will be your RNA, sir. And I hope you people know the full form very well. What is DNA, guys? DNA will be deoxyribose nucleic acid, and RNA will be ribose nucleic acid. Okay. DNA will be deoxyribose nucleic acid, and RNA will be ribose nucleic acid. Okay. Guys, you people are actually posting doubts. I'll take the doubts at the end of the class. Okay. Or else do one thing here. Please post your doubt in the group. In the group, I'll keep on answering. No problem with that. In the WhatsApp group. Okay. Chalo, no problem. First of all, let's get started with the class today. Okay. So nucleic acids will be of two categories in our body, so DNA and RNA. DNA stands for deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. Name itself is telling you that it is a nucleic acid. Name itself is telling you that it is a nucleic acid, sir. Deoxyribose and ribose. And first we are going to learn about the DNA right now, guys. So let's get started with the DNA. Okay. चलिए सर स्टार्ट करते हैं. So in DNA, first we are actually going to learn about the structure of DNA. I hope each and every student knows that a DNA has got two strands. It's actually a double stranded structure. There will be two strands and it will be helical. है ना? A DNA has got like two strands and it will be helical. Okay. Wow, सुभाष, क्या ज़बरदस्त डाउट पूछा आपने. एंजाइम इज इम्पॉर्टेंट सर. ओ भाई साहब, बायोकेमिस्ट्री इज ऑल अबाउट एंजाइम्स ओनली. Too much है. It is too much. Again the same thing. पूरी रामायण होने के बाद में रामा कौन था, सीता कौन था. Rate limiting enzymes important. Enzyme deficiency leading to diseases. Enzymes important. So it's biochemistry is only about enzymes only. And today I have sent you the LMR. Have you seen that last minute revision? In that all the enzymes I have given you at one place. Just keep on revising that. Okay. So chalo. DNA is made up of how many strands, sir? Two strands, and it is helical. Everyone please pay attention here right now, guys. Imagine that this is like one strand of the DNA and this will be like another strand of the DNA. So it is like double stranded. Okay. And the double stranded DNA is actually helical, twisted around each other. Fine. Now let us go a little bit more in depth. अब मुझे और depth में पढ़ने का मन कर रहा है कि sir what is this each strand made up of? And now in this each strand it is made up of something known as nucleotide. Nucleotide. Okay. Each strand is made up of nucleotide. Nucleotide. Okay. No issues, Shubhas. Okay. Take it easy. Chill. So DNA is made up of how many strands, guys? 2 strands. And what is that each strand made up of? Something known as nucleotide. So it is made up of many nucleotides. So please write down there. The 2 strands of DNA are made up of many nucleotides. Polymer of nucleotides. Okay. So DNA is made up of 2 strands and each strand is a polymer of nucleotide. Now obviously what is our next doubt in our mind? Sir, what is this each nucleotide made up of? Remember, the each nucleotide is made up of like 3 things. Number 1, it is made up of a sugar molecule. And number 2, it is made up of a phosphate group. Okay. And number 3, it is made up of a nitrogenous base. Sugar, phosphate and nitrogenous base. So I'm going to start this topic again from the scratch as you are seeing here right now, guys. So please take the advantage of that and learn properly. And later on, जैसे मैं आगे बढ़ता जाऊँगा, we are going to study higher topics like replication, transcription, translation and all. And you can understand those topics only if you are perfect in this one, guys. So a DNA is made up of how many strands, sir? 2 strands. And in that 2 strands, each strand is made up of many nucleotides. And each nucleotide is made up of sugar, phosphate and nitrogenous base. Ab chaliye sir, let us go in more depth and learn about all the 3 of them one by one, sir. The sugar, phosphate and nitrogenous base, sir. Okay. Now can anyone tell me which sugar do you have in the DNA? See, sugar is the another name that is used for carbohydrate. Carbohydrate common name will be what, sir? Sugar. And which sugar do you have in DNA? It will be deoxyribose sugar. Oh my god. It will be deoxyribose sugar. No need to even learn the name of this one. Already the name is there in the DNA full form only, sir. DNA full form is deoxyribose nucleic acid. So that itself is a sugar. Now this deoxyribose sugar will have how many carbons, guys? 5 carbons. And now draw the structure along with me. Oxygen here and then there will be like 4 carbons here. And the 5th carbon will be here, sir. Carbon number 1, carbon number 2, carbon number 3, carbon number 4 and the carbon number 5. So this diagram should be like printed in your mind, sir. Printed. This is going to help you till the end. Carbon number 1, carbon number 2, carbon number 3, carbon number 4 and carbon number 5. This is ribose sugar. And now till now you're expert. D means removal and oxy means oxygen, removal of oxygen. So from this sugar we are going to remove one oxygen from second carbon. Not required in that much depth in your MC exam. Take it easy. तो मतलब मान लो कि दिस इज द राइबोस शुगर एंड राइबोस शुगर हैज गॉट हाउ मेनी कार्बन, गेट 5 कार्बन. और यहाँ से हम क्या कर रहे हैं, एक ऑक्सीजन निकाल रहे हैं. इफ यू रिमूव वन ऑक्सीजन इट इज गोइंग टू बिकम व्हाट ओनली, इट इज गोइंग टू बिकम डीऑक्सी राइबोस शुगर. इट बिकम्स डीऑक्सी राइबोस शुगर. हो गया. तो विच शुगर डू यू हैव इन डीएनए? नो नीड टू मग अप गाइस, जस्ट यू नो द नेम ऑफ द डीएनए, यू आर डन. सो डीएनए फुल फॉर्म इज डीऑक्सी राइबोस न्यूक्लिक एसिड. So therefore this one is the deoxyribose sugar. Now out of these 3 things I am done with the sugar molecule, done. Now welcome to the phosphate group. Phosphate group, nothing to learn, sir. Phosphate group will be just PO4 minus 3. I hope you all know from our school days. PO4 minus 3 phosphate group. And what about the nitrogenous bases? We have to learn about the nitrogenous bases in detail. Welcome. Now please write on the heading in the center of the page as nitrogenous bases. Nitrogenous bases are in turn divided into 2 categories. One will be your purines, another one will be the pyrimidines. Purines and pyrimidines. And the purines are actually made up of 2 things. Number 1 purine will be your adenine and another purine will be your guanine. So one of the purine will be adenine, another purine will be guanine. And in pyrimidines, one of the pyrimidine will be the cytosine, another one will be the thymine. Okay, THYMINE, thymine. So purine will be adenine and guanine and pyrimidine will be cytosine and thymine. I think guys I don't have to repeat it again and again. Please learn along with me in the class. So this is लिखते जा रहे हो as you people are writing. So keep on learning along with me. Is it okay? Is it okay? So my dear friends, the purines are adenine and guanine and the pyrimidines are cytosine and thymine. ये तो सबको पता है. Now let me update your knowledge. Sir, everyone knows the purines, but remember, they will never teach you in our school days. These 2 are major purines. Adenine and guanine are major purine. यार अगर ये major purine होगा, definitely there should be minor purines also. Yes, you even have got the minor purines also. And what will be the minor purines, sir? Yes, right on there. Minor purines are xanthine, Number 2, hypoxanthine, and number 3 will be the uric acid. So xanthine, hypoxanthine, and the uric acid, these three are the minor purines. I will tell you the use of that later on in the class, don't worry. So now please keep on copying. Is it okay? So I repeat again, nitrogenous bases are again divided into how many categories, guys? 2 categories, purines and pyrimidines. The purines and pyrimidines, adenine, guanine, cytosine, thymine, and the minor purines will be xanthine, hypoxanthine, and uric acid. Okay? Done? Done. And now please welcome back to yes, this thing here. So you have to remember in order. I know everyone have learned about adenine, guanine, cytosine, thymine in your school days and college days and all. But my dear friends, I want you all to remember everything in sequence. Okay, I want you all to remember everything in sequence, proper order, not haphazard here and there, guys. So remember, DNA is made up of how many strands, sir? 2 strands. And out of the 2 strands, yes, remember, it's a polymer of, each strand is made up of many nucleotides. And what is that each nucleotide made up of in turn? Sugar, phosphate, and nitrogenous base. Which sugar? Deoxyribose. Phosphate, PO4 minus 3. And which nitrogenous base? Nitrogenous base are purines and pyrimidines. Purines are adenine and guanine. Pyrimidines will be cytosine and thymine. Okay, bacho? So that is like introduction. And now do one thing here. Again write down that in the center of the page as separately nucleotide. And in the bracket write down there will be a sugar, then phosphate, and then nitrogenous base. Okay. What I am trying to teach you right now, just now we have studied about all the 3 of them individually. We have learned about the sugar, we have learned about the phosphate, we have learned about the nitrogenous base, one by one. Individually we have learned. Okay, fine. But now let's see how they are attached to each other. Acha. Okay. So my dear friends, there will be a sugar molecule. And on this sugar molecule, carbon number 1, carbon number 2, carbon number 3, carbon number 4. And here there will be carbon number 5. Okay, ji. Sugar, done. Remember, always the phosphate group will be attached onto the carbon number 5. Phosphate hamesha carbon number 5. And nitrogenous base is always attached onto the carbon number 1. So now I'm having sugar, I'm having phosphate, I'm having nitrogenous base. I repeat again, I'm having sugar, I'm having phosphate, I'm having nitrogenous base. And from today onwards, from now onwards, in fact, this diagram should be like printed in your mind, sir. That's a nucleotide. The moment you listen the word nucleotide, that picture should come in your mind. Sugar, phosphate, nitrogenous base. Okay. Now with this, let us go to the next concept right now, guys. Nowadays in your exam, they are asking about something known as nucleoside. Now, sir, what is the meaning of nucleoside? Nucleoside will just have the sugar molecule along with the nitrogenous base. That's all. The sugar molecule along with the nitrogenous base, no phosphate group. I think you yourself can make the diagram right now. Please make the diagram, sir. Yes, there will be a sugar molecule, and on carbon number 1 there will be only nitrogenous base, no phosphate group. और इसको कहते हैं हम nucleoside. It is known as nucleoside. So there are many MCQs coming based on this one, guys. Okay, that is nucleotide and the nucleosides. Nucleotide और nucleotide पर बहुत सारे सवाल आ रहे हैं. ठीक है, can you manage this? Can you manage this now? Yes. हो गया. And now the next one, guys. Make a table with 3 columns. अब देखो sir, now conceptually, let me teach you the USMLE pattern, sir. Conceptually, in the very first column, we are going to write down the name of nitrogenous bases. Okay, you already know the name of the nitrogenous bases. What are nitrogenous bases? Purines and pyrimidine. What is purine? Adenine and guanine. What is pyrimidine? It will be cytosine and thymine. Okay, we know this one. So first column already we know. Welcome to the second column. Now what you do is take the nitrogenous base and attach to sugar. Oh my god. My dear friends, can you please tell me what is this molecule now? Come on, tell me what is this molecule right now? Nitrogenous base with the sugar. Nitrogenous base with the sugar will be nucleoside. Are you getting my point here? That's a nucleoside. Very good. So, my dear friends, my dear doctors, yes, this is the sugar molecule here, and on the carbon number 1, nitrogenous base. Now listen here, sir. There are many chances of getting confused here right now. So 200% concentration here right now, guys. Out of these 4 nitrogenous bases, adenine, guanine, cytosine, thymine, I am taking the first one, adenine, and I am going to attach adenine here, sir. Where? In the place of nitrogenous. Bolo bacho, this is adenine with the sugar. What is this in general? Nucleoside. And what is the name of this one? It is now known as adenosine. Oh my god. Are you getting my point here? Students, many of them, 99% of the students don't know what is the difference between adenine and adenosine. Adenine is just a nitrogenous base, whereas adenosine will be nitrogenous base with the sugar. That means it is a nucleoside. Are you getting the difference now? I hope my students have a clarity right now, sir. So adenine with the sugar will be adenosine. अगर मान लो we have attached guanine here, sir. अगर मान लो हमने यहाँ पर guanine लगा दिया, तो guanine with the sugar will be, yes, it will be guanosine. Guanosine. Okay. Suppose if you attach cytosine there, sir, now the name will become cytidine. And number 4, yes, it will be thymidine. Wow, very good. Okay, so I hope like now you people are having a clarity between the nitrogenous bases and the nucleoside, guys. Are you clear with this one? Now welcome to the last and final column right now. Take the nitrogenous base from the first column, attach that with the sugar. It becomes what? Nucleoside. अब उसको phosphate लगा दो. It becomes what? It becomes nucleotide. Lovely, crystal clear, conceptual. Are you understanding your point here right now? तो देख रहे हो sir, like how conceptually we are studying here. First we have taken nitrogenous bases. Okay, these are the names of nitrogenous bases: adenine, guanine, cytosine, thymine. Then attach the nitrogenous base with the sugar, nucleoside: adenosine, guanosine, cytidine, thymidine. And now attach that nitrogenous base and sugar with the phosphate group. It becomes nucleotide. And now, my dear friends, look at the magic. Look at the magic here. Hello everyone, please stop writing and look at my screen here. Stop writing and look at my screen. This one will be the sugar molecule. I've attached adenine. Adenine with the sugar. Adenine with the sugar. Adenosine. Adenine with the sugar. Adenosine. Come on. Adenine with the sugar, adenosine. If I attach a phosphate group here on carbon number 5, wow, adenine with the sugar, adenosine with one phosphate, adenosine monophosphate. If I attach one more phosphate group here, this is nothing but adenosine diphosphate. If I attach one more phosphate group here, it's nothing but adenosine triphosphate. Oh my god. ATP, ATP, ATP. हम लोग LKG UKG बचपन से रट्टा मार रहे, रट्टा. This is nothing but your ATP, sir. Adenine with the sugar, adenosine with the phosphate group, adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. Adenine with the sugar, adenosine, and with 3 phosphate ATP. Oh, that means ATP is an energy rich molecule as well as it's a nucleotide. It's a nucleotide. So how can you tell it is a nucleotide, sir? Yes, you can clearly see. Do you have sugar? Yes. Do you have nitrogenous base? Yes. Do you have phosphate? Yes. It's a nucleotide. Understanding my point here right now? तो लिखो सर, adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. These are all nucleotides. Adenosine monophosphate, diphosphate and triphosphate, all these are nucleotides. And similarly, if it is guanine, guanosine monophosphate, guanosine diphosphate, and then guanosine triphosphate. And if it is cytidine monophosphate, cytidine diphosphate, and cytidine triphosphate. And if it is thymine, thymidine monophosphate, thymidine diphosphate, and thymidine triphosphate. That will be the nucleotides, and that completes your basics. हो गया बच्चों? हेलो, okay, nitrogenous base structure not required, Divya. Whatever I'm teaching you, just learn that. They will never ask you the nitrogenous base structure. Are you all done? Now in this, what I have completed is like, nitrogenous base, nucleoside, nucleotide, everything has been done. And if you're done with this one, now you people are ready to study the structure of DNA. Structure of DNA पढ़ने के लिए आप लोग तैयार हो. So please now write down the heading as structure of DNA. Please write down there as the structure of DNA, sir. Structure of DNA. And I hope you all know that the structure of DNA was given by Watson and Crick. Yes. So therefore it is known as Watson and Crick DNA. The name of the DNA itself is Watson and Crick DNA. Watson and Crick DNA. And this Watson and Crick DNA is also known as BDNA. इसको ना BDNA भी कहते हैं. So first of all, write down the name of the DNA. We are going to learn the structure of the DNA. Which DNA, sir? Watson and Crick DNA. That is also known as BDNA. And now, okay. Listen the planning here right now, sir. Planning तो इतना आसान है. पहले हम लोग ना एक strand बनाएंगे. We are going to make one strand. One strand is a polymer of nucleotides. Then we are going to make another strand of the DNA, sir, and there is also a polymer of nucleotide. Then join them together, sir. क्या बात है? ओहो, खतरनाक planning. Okay, so one strand of DNA, another strand of DNA, and then हम दोनों को जोड़ देंगे, sir. Okay, one strand, another strand, दोनों को जोड़ देंगे. ठीक है जी? Okay. तो पहले एक स्ट्रैंड बनाएंगे, फिर दूसरा स्ट्रैंड बनाएंगे, दोनों को फिर जोड़ देंगे, फिर बाद में मोड़ देंगे. We'll make them helical. Okay. तो चलिए सर, लेट्स स्टार्ट करते हैं. शुभ आरंभ. Now what is that one strand of DNA made up of? It is made up of many nucleotides. Sugar, attached to that will be the phosphate group, and then adenine, nitrogenous base. Now what is this, guys? Nucleotide. But this is only one nucleotide. It is made up of many. Let us take many nucleotides. Sugar, and then thymine, and then phosphate group. Sugar, and then guanine, and then phosphate. I'm just taking randomly, sir. Okay. Sugar, and then adenine, and a phosphate group. And then मान लो कि sugar, then cytosine and phosphate. Just something randomly. It doesn't mean to be in the same order. My main intention is, yes, we are going to take multiple nucleotides. We are going to take many nucleotides, polymer of nucleotide. Now after taking the many nucleotides, what is our next challenge? Our next challenge is to join them together. How they are joined together? Remember, the sugar of one nucleotide joins with phosphate of another nucleotide. Sugar with the phosphate, sugar with the phosphate, sugar with the phosphate. Sugar of one nucleotide joins with the phosphate group of another nucleotide. Yes, you can see one chain is formed there, one strand is formed there. Before moving ahead, yes, there is a bond formed here. So whenever there is a bond formed, there should be some name to the bond, right? What is the name of this bond? This bond will be phosphodiester bond. इसको हम कहते हैं phosphodiester bond. Now this phosphodiester bond, is it a strong bond or a weak bond? Remember, it's a very strong bond. Phosphodiester bond, it's a very strong bond. Okay guys, what is the meaning of this strong bond here right now? If you increase the temperature or if you increase the pH, अगर आपने temperature बढ़ाया और pH बढ़ाया, remember, will this bond break? No, this bond will not break. That's important. So I'll tell you the importance of this one later on. अभी के लिए note कीजिए इसे. Phosphodiester bond, name of the bond, and it's a very strong bond. And what is the meaning of the strong bond, guys? If you just simply increase the temperature or pH, this bond is not going to break. Okay. And now, my dear friends, the last and final point. In this one strand of DNA, one of the end will be 5 dash, another end will be 3 dash. One of the end will be 5 dash and 3 dash. How will you decide? Look at that one strand. At one of the end will be having the phosphate group, free phosphate group. The other end will not have a free phosphate group. So whatever end is having a free phosphate group, जहाँ पर भी free phosphate group होता है, attached on which carbon? 5th carbon. That is 5 dash end. And the opposite end will be obviously 3 dash. Lovely. So we are done with everything possible here in one strand. चलिए. What is our planning? Our planning is we need to make one strand and then we have to make the another strand of DNA, है ना? तो चलो एक strand बन चुका है, अगला strand बनाते हैं. But to make one more strand here right now, guys, we are not ready for that. Let us first of all learn some basics and then come back. So you do one thing in your books, don't draw anything on the right side here. Don't write anything on the right side, write below the diagram. Let us learn some basics and come back. And the basics you need to learn about the Chargaff's rule. Okay. Let us see what is this Chargaff's rule says. Number one, Chargaff's rule says that the number of purines are equal to number of pyrimidines. Number of purines are equivalent to number of pyrimidines. ठीक है ना? Now what are purines, guys? Adenine and guanine. What are pyrimidines? Cytosine and thymine. If you add them, total number of purine. If you add them, total number of pyrimidine. And according to Chargaff rule, it says that these two are equal, it seems. So that person has done some research. तो मानना पड़ेगा. Don't ask me how, why. Otherwise, you also go and do the research, and again you will be getting the same answer. Okay. So why not agree now? So Chargaff's rule says that number of purines equals to number of pyrimidine. Adenine plus guanine equal to cytosine plus thymine. Number two, second rule of Chargaff is actually about the complementary pairing. It says about the complementary pairing. Now what is this complementary pairing, sir? Purines, adenine and guanine, pyrimidines, cytosine and thymine. And after doing the research, that person has found that adenine always loves to bind with thymine. Adenine हमेशा thymine से bound होता है, that too with a double bond. And guanine always loves to bind with cytosine. So guanine always loves to bind with cytosine with the help of a triple bond. So adenine with thymine and guanine with pairings. So Chargaff's rule, it says that number of purines equivalent to number of pyrimidines. And it is saying about the complementary pairing. Complementary pairing is like ATGC. Is it okay? Is it okay? Come on, tell me. Very good. Now once you have learned this Chargaff's rule, अगर आपने एक बार ये Chargaff's rule पढ़ लिया है sir, and now you people are ready to learn or ready to make the another strand of DNA, sir. So let's make the another second strand of DNA. Now just 1 second, look at the screen here right now, guys, everyone here. Another strand of DNA is also made up of what? Only nucleotides. So there will be sugar molecule attached along with the nitrogenous base here, that is thymine. But this time phosphate group will be attached complementarily, opposite. Don't attach above, complementary, opposite. Come on. Sugar, adenine, phosphate group. Sugar, cytosine, phosphate group. Sugar, thymine, phosphate group. And then sugar, guanine, phosphate group, sir. Okay. Now we need to join them together, and you already know. Sugar will be joining with the phosphate group. Sugar with the phosphate. Sugar with the phosphate. Sugar with the phosphate. And you know everything about that. That is a phosphodiester bond, strong bond, time pass. Now please decide which end will be 5 dash and which end will be 3 dash, sir. Yes. Here I am able to find the free phosphate group. Lovely. That is a 5 dash end, and this end will be 3 dash. Oh my god. That is the reason why your DNA is said to be anti-parallel. One of the strand of DNA will be 5 dash to 3 dash. The other strand of the DNA will be 3 dash to 5 dash. One of the strand is 5 dash to 3 dash. Another strand is 3 dash to 5 dash. Are you understanding now? At least today, are you understanding in your life? That is why DNA is known as anti-parallel. Anti-parallel. Okay. Now that is one strand of DNA. There is another strand of DNA. Now let us join them together. And how they are joined together? कैसे? Adenine loves to bind with thymine, double bond. Thymine, adenine, double bond. Double bond. Cytosine, guanine, triple bond. And how do you know that? Who said you that one? चारगव भाई साहब. चारगव भाई साहब ने कहा है कि ऐसे ज्वाइन होते हैं. ठीक है जी. So we are applying Chargav's rule here. Whatever it is, double bond or triple bond, my dear friend, there should be some name to this bond, right? Yes. These bonds are known as the hydrogen bond. Hydrogen bond is a strong bond or a weak bond? Weak bond. What is the meaning of weak bond? Increased temperature or increased pH, and these bonds are going to break apart. अगर आप टेम्परेचर बढ़ाओगे या pH बढ़ाओगे तो क्या हो जाएगा? ये बॉन्ड ब्रेक हो जाएगा. ठीक है. That's all. And now you can clearly see that our double stranded DNA is completely formed here. Take this DNA and fold it, make it helical, sir. That becomes your DNA, sir. DNA structure हो गया, such an easy thing. Unnecessarily students will be confused, sir. Hardly 5-10 minutes हो गया, sir. Are you done? Come on, बच्चों. Are you done? Very good. That means here, what is the bond that is present between the nucleotides in same strand? In same strand there is a phosphodiester bond, sir. Okay. And then what is the bond between the two nucleotides in different strand? Hydrogen bond. So please be very careful. We are having like two bonds here right now. In same strand there will be phosphodiester bond, and in two different strands there will be hydrogen bond. Okay na? Welcome Tino, welcome Anil. Okay, thank you for understanding. Okay, तो दोनों न्यूक्लियोटाइड्स के बीच में between the two nucleotides in same strand phosphodiester bond, in two different strands it will be hydrogen bond, sir. हो गया डन. चलो बच्चों आगे बढ़ते हैं. And if you are observing properly, I'm done with DNA structure, I'm done with the bonds, I'm done with the weak bond, I'm done with the strong bond, टेम्परेचर, pH, आई एम डन विद चार्गव रूल. तो एक ही जगह पे हमने बहुत सारे टॉपिक्स कंप्लीट कर दिए. एवरीथिंग डन 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 सर. चलो अब हम बढ़ते हैं आगे. आई होप पीपल आर एंजॉइंग. तो लेट मी गिव यू अ क्वेश्चन हियर राइट नाउ सर. इन अ डीएनए, लिखो बच्चों, लास्ट दिसंबर एग्जाम क्वेश्चन. इन अ डीएनए इफ adenine is 30%, then guanine will be how much? Adenine is 30%, then guanine is how much? 60, 20, 20, 60, पता नहीं क्या क्या आंसर. ओके सर, यहाँ देखो सर. Let me tell you the answer for this one here right now, guys. Just now I told you in a DNA what is it applicable to Chargaff rule, and Chargaff भाई साहब ने क्या कहा था? Adenine loves to bind with thymine and guanine loves to bind with cytosine. Okay, everyone look here. No need of formula, nothing. कोई formula का जरूरत नहीं, कुछ नहीं सर. Common sense, दिमाग लगाओ. In a DNA, Chargaff's rule applicable. Adenine loves to bind with thymine, guanine with cytosine. Adenine is how much? 30%. Thymine should be how much? 30% only. Why? Because it is binding with that one. 30 and 30 how much? 60%. If this is 60%, the remaining should be 40%. And if the remaining is 40%, this is 20% and this is 20%. Wow. And what is asked in the question? Guanine. Guanine is 20%, sir. Ha. Understanding here right now? No need of formula, nothing, sir. And why we are taking it like this? We assume our DNA to be complete 100%, sir. Apna DNA perfectly complete, sir, 100%. In a DNA. Okay? Are you done? Very good. This is how you have to solve the problem. Already these kind of problems are given in the exam. Chalo, if your people are perfect, let me give one more question, sir. In a DNA, if cytosine is equals to 65%, then tell me what about the thymine? Thymine कितना होता है? अबे Titan, you are reading the notes or what? बैठ के नोट्स सामने रखे नोट्स पढ़ रहे हैं. हेलो, हो गया बच्चों? ना, oh my god, oh my god, oh my god, 35, 17.5, 130%. अरे रे रे, अरे रे, अरे रे, अरे रे। चलो सर, अब यहाँ पर देखो सर। Now everyone here right now, guys, everyone here right now, let me tell you the answer. Adenine loves to bind with thymine and guanine loves to bind with what, sir? Cytosine. What is given here in the question? Cytosine here. How much is cytosine? 65%. How much is guanine? 65%. 65 कितना होता है? 130%. Not possible. I want my DNA to be 100%. It cannot be more than 100%. Possible ही नहीं है. So what is the answer? The answer for this one will be this type of DNA does not exist. And don't take it easy. It was given in the exam, sir. Answer was in this type of DNA does not exist. This type of our DNA has to be perfect, sir. 100%. It can't be more than 100%. So okay. So this is already given in the MCA exam. Take care. So these are like two sample questions. This type of question can come in the exam. Take care. Okay, MacBook. चलो आगे बढ़ते हैं. Let's go ahead. And now, my dear friends, one we are done with the structure of DNA. We are done with the structure of DNA. Then we are done with the Chargas rule, and we are done with the problems based on Chargas rule. And now some small topics. Now in the small topics, yes, write down there topic number 1, that is types of DNA. So we are done with the structure of DNA. Now please write down the heading as types of DNA. There are totally 6 varieties here: A DNA, B DNA, C DNA, D DNA, E DNA, Z DNA. Someone was asking me what is this B DNA, sir? I think you'll understand. There are a variety of DNA, sir. There are types of DNA: A DNA, B DNA, C DNA, D DNA, E DNA, Z DNA. And for your MCA exam, you have to always remember the comparison between the B DNA and Z DNA, sir. So please do one thing here. Write down on one side as B DNA. Please write down on one side as B DNA, and on the other side you write down as Z DNA, sir. Z DNA. What is the another name of B DNA? B DNA is known as the Watson and Crick DNA. Okay. ठीक है. Now my dear friends, remember B DNA double stranded, Z DNA double stranded. Okay. B DNA is also helical. This one also helical. But now welcome to the difference. B DNA is having a right-handed helix, and whereas Z DNA will be having the left-handed helix. Okay, so I think you already know helix. I can make it like this also. I can even make it like this also. Okay, na? So it can be right-handed helix or it can be a left-handed helix. So therefore, the only one point that you have to remember here will be B DNA will be having the right-handed helix, whereas the Z DNA it will be having the left-handed helix. This topic is done, sir. Structure of DNA हो गया, and then we are done with the types of DNA. Should I remove this? Come on, sir, be quick. Come on, be quick. Now, my dear friends, everyone observe here right now, sir. Next topic. थोड़ा ना ध्यान से देखना. For example, I'm having one strand of DNA, another strand of DNA, and I have joined both the strands of DNA like this, sir. Suppose if you are increasing temperature, suppose if you are increasing pH, either one of this, sir, either increasing temperature or increasing pH. Tell me which bonds will break. अगर मान लो कि हमने टेम्परेचर बढ़ाया, pH बढ़ाया, कौन सा बॉन्ड ब्रेक होता है? Yes, the hydrogen bonds in the middle will break. And if the hydrogen bonds are going to break in the middle, what is going to happen to both the strands? Both the strands of the DNA will separate apart. And both the strands of DNA will separate apart. This phenomenon or this concept is known as denaturation of DNA. What is it known as, sir? Denaturation of DNA. So therefore, write down the next topic. Heading likho, sir. Denaturation of DNA. It is also known as melting of DNA. Now there are two MCQs possible here, sir. MCQ number 1. Denaturation of DNA कैसे होता है? How it will occur? By increased temperature or decreased temperature? Increased temperature. Increased temperature or decreased pH? Increased pH. Denaturation of DNA, how it will occur? By increasing temperature or by increasing pH. And then second question possible, which bonds will break here? Yes sir, we all know that the hydrogen bonds will break here in this denaturation of DNA. So the hydrogen bonds are going to break in denaturation of DNA. Now hello my dear friends, everyone here, just look at me here sir, 1 second. सब लोग मुझे यहाँ पर देखिए. मान लो कि these are the two strands of DNA. Temperature बढ़ाओ, pH बढ़ाओ, both the strands will separate apart. Denaturation. Bring the temperature again back to normal, decrease temperature. If you decrease temperature or decrease pH, both the strands of DNA will again come back and coil, and that is known as renaturation of DNA. So please write down there as renaturation of DNA. Renaturation of DNA is also known as annealing of DNA. And I will always learn like smartly. I'm not going to learn it again. Okay, decrease temperature, decrease pH and confuse. Just write down there vice versa. It's opposite of denaturation. So always study only one thing, the other thing will automatically follow. Okay. So denaturation of DNA or melting of DNA by increased temperature. Obviously renaturation will be by decrease in temperature. Okay, bachcho? Are you all done? Sure. Now if you are done with this topic, now welcome to the last and final topic sir in this one, in this chapter. Yes, organization of DNA. Organization of DNA, and this is something very much important nowadays for our exams. ठीक है ना. हाइड्रोजन बॉन्ड्स फिर से फॉर्म होते हैं सर. हाइड्रोजन बॉन्ड का क्या होता है, मतलब क्या, ब्रेक होते हैं, फॉर्म होते हैं, बॉन्ड्स है, दिल अगेन ब्रेक, दिल अगेन फॉर्म. Welcome to the organization of DNA. Let us see the arrangement of DNA. And now 200% alert. I will take you like in depth right now sir. चलिए सर मेरे साथ साथ में let's go in depth. मान लो कि ये वाला आपका सेल है, मान लिया. चलिए सर सेल के अंदर चलते हैं. Inside the cell you'll be having nucleus here. मान लिया. Inside the nucleus you'll be having the fine thread-like structure that is known as chromatin material. Okay. And we are now going to see it microscopically. चलिए से देखते हैं. Today I am going to take that chromatin material out. Okay, no problem. There is a thread-like structure known as chromatin. But like you people and you know me and you people are like very much interested in studying, है ना? You people are like very much interested in studying, and you people are like, you know, sir, I don't want to see only microscopically. I want to see ultra microscopically. खाली बस दूर से नहीं देखना है सर. I don't want to just see, okay, from very far. मुझे ना बहुत करीब से देखना है, ठीक है ना. Okay, let us see from pass. So when you see this chromatin material बहुत करीब से, yes, you will be able to see something more details, है ना. Common sense. Every time we look from very far, then when we go very much nearer, more details we can see. So similarly in the chromatin material also we can see more details, and that will be, yes, at regular intervals you'll be having beads. Oh my god. So जो हम दूर से देख रहे हैं, when you are looking at the chromatin material from very far, it is just a thread-like structure. But when you see it more closely, they are at regular intervals, they are like bead-like structure. And now these bead-like structures I am really interested about. कि sir क्या है? So remember, first of all, the name of this one, guys. These bead-like structure are known as nucleosomes. क्या कहते हैं? Nucleosomes. And now I want to see this nucleosome more closely. What is that? So my dear friends, what is that nucleosome? You know, in the center of the nucleosome there will be a histone protein, and on that histone protein our DNA will be coiled. So in the center there will be a histone protein. Let me draw the histone protein with another color, blue color. Histone protein, histone protein. And on that histone protein, what is going to happen? Our DNA will be coiled. Acha. Something like this, understanding? And this is giving the appearance like a bead, like a nucleosome. Okay. Understanding? तो इस तरह से हम उसको ना zoom करते चले जाएंगे. Keep on zooming. So my dear friends, let's go first of all inside of a body. देख रहे हो sir? Like, are you understanding? So inside of a body, bead, Srinivas, bead. By the way, don't even remember bead, remember nucleosome. What is the name of that one? Nucleosome. So my dear friends, inside the cell, nucleus, inside the nucleus, fine thread-like structure, chromatin. And chromatin, look at that closely, every regular interval, bead-like structure known as nucleosome. Now, what is that each nucleosome made up of? That is the question nowadays coming. Remember, in the center, you'll be having histone protein. How many histone proteins will be there in the center? 8. That is the reason why you can write down as octamer of histone proteins. Octamer of histone protein. Madhyalo, in the center, you will be having octamer of histone protein. What is the name of this histone protein? Okay, H2A, H2B, H3, H4. Sir, you said that there will be totally 8 histone protein. You are giving the names of only 4 of them. Remember, there will be 2 copies of each of them. So there will be 2 copies of H2A, H2B, S3 and H4. There will be 2 copies each of H2A, H3, H2B, S3 and H4. Sir. And surrounding that octamer of the histone protein, yes, you will be having, yes, coiled by DNA. DNA will be coiled surrounding that. Any problem here? And this one will be known as, like one nucleosome, and this is like another nucleosome, something like this. And now, my dear friends, if you are observing the names of the histone proteins, the names of the histone proteins are actually starting with H2. There is something H2A, there is something H2B, there is something H3 and H4. Sir, where is H1? H1 कहाँ है? यहाँ देखो. This is nucleosome and this is another nucleosome. Nucleosome and nucleosome. Between them, they are connected with the help of this DNA. And what is the name of this DNA? This DNA, thank God, doctors have named it as linker DNA. Nucleosome, okay, they're linked by linker DNA, and this linker DNA is actually associated with the histone protein number 1, H1. So please write down the statement there, sir. H1 histone protein is associated with linker DNA. I repeat the statement again, write down. H1 histone protein is associated with linker DNA. H1 histone protein is associated with linker DNA. I repeat again, H1 histone protein is associated with linker DNA. Are you all done? Okay. So let us just do the revision once here right now, guys. Everyone here right now. So this is a cell here. Inside the cell nucleus. Nucleus will have chromatin. Chromatin, look closely. Bead-like appearance. Nucleosome. Nucleosome, look closely. In the center, you'll be having octamer of histone protein, and it is made up of H2A, H2B, H3, H4. H1 is along with the linker DNA. Now, my dear friends, can anyone tell me what is the charge on the DNA? DNA is positively charged or negatively charged or neutral? DNA is positive, negative or neutral? Yes, welcome here, sir. Let's zoom that diagram. So, थोड़ा और zoom करते हैं. Histone protein, and onto that histone protein, our DNA will be coiled. Remember, our DNA will be negatively charged. Why? I told you already today only DNA will be having nucleotides, and nucleotides will be having phosphate group, sugar, phosphate group and nitrogen. What is the formula of phosphate group? PO4 -3. Because of that phosphate group, DNA will be negatively charged. DNA will be negatively charged. What about the histone protein? Histone protein will be positively charged. है ना? And the question in your exam was, the histone proteins का charge क्या होगा? It will be positively charged. Why? Remember, the histone proteins are positively charged because of more of, number 1, that is your lysine amino acid. Number 2, that is arginine amino acid. So it's a protein, and protein is made up of what? Amino acid, and it is having excess or more of lysine and arginine. So lysine and arginine, more of that will make it more positive in nature. So histone protein will be positively charged, and the DNA surrounding that will be negatively charged. I hope you are done with this one here, and this completes your first topic. We are done with the first topic for today. Nucleotide, nucleoside, structure of DNA. Okay, and then we are done with the denaturation, renaturation. We are done with types of DNA and arrangement, organization of the DNA. That completes the first topic. I'll give you like 2 minutes of time, and then we'll start with the second topic. So hello everyone, welcome back now. I think you are done with the revision part. First topic done. Let's start with the second topic for today. Come on guys. The second topic that we are going to begin for today will be heading Licosa, nucleotide metabolism. Please write down the heading there as nucleotide metabolism. So we are done with the carbohydrate, protein and lipid metabolism, and now this is nucleotide metabolism. It's an easy topic, sir. I think you're observing the trend. The first topic that we have learned, carbohydrate metabolism, was the biggest one. Then protein, a little bit smaller one, and lipid metabolism yesterday it was done in 1 hour. So it is the smallest one. And nucleotide metabolism, welcome sir, 40-45 minutes this will be done. Okay. Now nucleotide metabolism, the first thing that we need to learn here is about the purine ring, sir. We are going to learn about the purine ring, and we all know what are purines, guys. Purines are adenine and guanine. The first point to remember is that the purine ring will be double ring structure. Okay. What is the meaning of purine ring? Purine ring will be a double ring structure. And guys, in this double ring structure of the purine, one of the ring will be actually hexagonal. Okay. And another ring will be the pentagonal. So one of the ring will be like hexagonal, another ring will be like pentagonal, sir. So purine ring will be a double ring structure. It will be hexagonal and pentagonal. Done. And that too, it will be like heterocyclic, sir. Heterocyclic. Now what is the meaning of homo? Homo means same, and hetero means what's the different. So it is not made up of same atoms, it is made up of different atoms. For example, here you'll be having like nitrogen here, and then there'll be one more nitrogen here, one more nitrogen over here, and then there'll be one more nitrogen here. Okay. And everywhere else you'll be having carbon. Okay, so there'll be carbon, 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 carbon and carbon. So that is the reason why it is known as heterocyclic, made up of different atoms. Heterocyclic, it is made up of different atoms. And now when it comes to the numbering will be always given here in the anti-clockwise direction here right now, sir. Starting from here 1, 2, 3, 4, 5 and 6. And here it will be like 7, 8 and 9. Is it okay? And now if you ask me really, as a doctor, you don't have to learn the structures. ठीक है ना, ये जो chemical structures जितने भी होते हैं ना, वो पढ़ने का आपको ज़रूरत नहीं है. No need to learn the chemical structures. Is it okay? As a doctor, I don't know why in the foreign universities they'll make you learn all the structures and all those things. Structures not all required. And now also I'm telling you the purine ring structure is not required. Structure आपको ज़रूरत नहीं है. ठीक है ना? The structure is not required. Now the only thing that you require here is that you don't require to learn about the structure, the double ring. Don't worry about carbons and nitrogen. Don't worry about the numbering. Don't ask me, sir, why is numbering even in anti-clockwise? Why not clockwise? There is a rule. There is a protocol. I think you have heard about IUPAC nomenclature. So according to that nomenclature, the numbering, everything is given. But as a doctor, what should be my headache? My headache should be only which nitrogen and which carbon is given by which substance. कौन सा वाला नाइट्रोजन, कौन सा वाला कार्बन, कौन सा सब्सटेंस यहाँ पर कोऑपरेट करता है. ठीक है ना. सो नाउ लेट अस सी द नाइट्रोजन नंबर 1 राइट नाउ. रिमेंबर नाइट्रोजन नंबर 1 इज गिवन बाय aspartate. This is what you have to know as a doctor. Nitrogen number 1 is given by aspartate. Carbon number 2 and then the carbon number 8. These two are given by tetrahydrofolate. Tetrahydrofolate. And then nitrogen number 3 and nitrogen number 9. These two are contributed by glutamine. Glutamine. And recall my day before yesterday's class, or I think yesterday's class only. Carbon number 4, carbon number 5 and nitrogen number 7, this will be contributed by glycine. You remember yesterday's class? First topic only, aliphatic amino acid, glycine. And in glycine, one of the function I made you write down, it is helping in the formation of purine ring. Purine ring formation में help करता है. Carbon number 4, carbon number 5 and nitrogen number 7. These are contributed by glycine. And finally, carbon number 6 is given by carbon dioxide. Is it okay? So in the nucleotide metabolism, the first thing that you have to remember is actually the purine ring and the contribution here right now, sir. THF, someone wants the full form of THF here right now. It will be tetrahydrofolate. I think we have studied about this yesterday also. Tetrahydrofolate, dihydrofolate and all. So my dear friends, in the nucleotide metabolism, first topic and a small topic that you have to learn is about the purine ring, and you have to remember the contribution. तो एक बारी ना फिर से repeat करते हैं, everyone along with me. Nitrogen number 1 is given by aspartate, carbon number 2 and carbon number 8 is given by tetrahydrofolate, and then nitrogen number 3 and 9 are given by glutamine, and then carbon number 4, carbon number 5, nitrogen number 7 is given by glycine, and then carbon number 6 is given by carbon dioxide. ठीक है, I will tell you what is the use of this one within few minutes. Okay, तो चलिए हम आगे बढ़ते हैं. Now my dear friends, this topic is so simple that we are going to learn only two things here: the synthesis of purine and degradation of purine, purine formation and purine breakdown. That's all. If you learn that, that is more than enough for your MC exam, sir. So everyone here right now, please write on the heading as synthesis of purine. And as usual in every topic, first of all I am going to give you what guys, overview. मैं ना सबसे पहले आपको पढ़ाऊंगा overview से. Synthesis of purine will be done by two methods. It is just the overview, sir. One will be the de novo synthesis of purine. Another one will be the salvage pathway. So one is the de novo synthesis of purine, another one is the salvage pathway. There are two methods for synthesis of purine, sir. Okay, fine. And now my dear friends, what are the starting substance here? So first of all, let us discuss about de novo synthesis. The starting substance here will be the ribose 5-phosphate. And this ribose 5-phosphate will undergo certain reactions. After that it is going to form IMP. And this IMP is going to form what, sir? Your AMP and your GMPs. That's all. Okay. Don't think that the entire process is done, sir. We are writing overview only. So in de novo synthesis of purine, yes, remember what is the starting substance? Ribose 5-phosphate. Then that is going to undergo certain reactions to form IMP, and IMP is going to form your AMP and GMP guys. Is it okay? And now, my dear friends, listen here carefully. Let us discuss about this in detail. I want you all to listen to this overview very carefully, guys. Why? Because in this overview itself, entire topic will be done, sir. In the overview itself, entire topic will be done. इसी में सारा topic class हो जाएगा, sir. So I want all of you to please look at this overview very carefully. Is it okay? So please observe here right now, my dear friends. The moment you look at this word here right now, sir, ribose 5-phosphate, are you able to recall something? Hello everyone. Ribose 5-phosphate, are you able to recall something? Yes, that is obtained from HMP shunt. You can go back and check, turn the pages back and check. I told you HMP shunt, there are two substances obtained. One will be NADPH, another one will be ribose 5-phosphate. And today you can see it is helping in the formation of adenine guanine, purines, and purines are helping in the formation of, yes, DNA and RNA. ठीक है? अरीश सर, इसमें से क्वेश्चन कैसे पूछते हैं भाई साहब? पढ़ते जाओ ना, पता चलेगा. Keep on learning, you will come to know. Okay, अरीश. ओ अरीश, ओ भाई साहब, पढ़ते जाइए, पता चलेगा. iPad C is asking, sir, repeat the last point. Last point was पढ़ते जाइए, पता चलेगा. यहाँ देखो, de novo synthesis of purines. De novo synthesis of purines. The starting substance is ribose 5-phosphate. Ribose 5-phosphate, and that ribose 5-phosphate is obtained from HMP shunt. Okay, iPad. Ribose 5-phosphate is obtained from HMP shunt. ठीक है, done. Now it is undergoing certain reactions to form IMP. Sir, in the very beginning of this class, I taught you about AMP, GMP, and I taught you about the CMP and TMP. You remember the table? You remember the table? Okay. So we remember about the AMP, GMP, everything. But what is this IMP, sir? चलिए सर मैं आपको बताता हूँ what is this IMP. Listen here right now. First of all, learn the full name. IMP actually stands for inosine monophosphate. Inosine monophosphate. अच्छा ठीक है. Then sir, what is this inosine monophosphate? चलो, everyone here right now. I hope you are all perfect with this diagram here right now. This is sugar. एंड मान लो कि यहाँ पर फॉस्फेट ग्रुप ऑलरेडी है ऑन द 5th कार्बन. सपोज इफ आई अटैच adenine here right now guys, then what is the name of this one? Adenine with the sugar, adenosine with phosphate, adenosine monophosphate. Suppose if I attach guanine here right now, it becomes what, sir? Guanine with the sugar with the phosphate, guanosine monophosphate. I told you just few minutes back, adenine and guanine are major purine. There are certain minor purine. Hypoxanthine, xanthine, uric acid, you remember? तो now attach hypoxanthine. अगर मान लो कि यहाँ पर आपने hypoxanthine यहाँ attach कर दिया, the moment you attach the hypoxanthine here, remember hypoxanthine with sugar is known as inosine. Hypoxanthine with sugar is known as inosine. So guys, I can't help you out here. Adenine with the sugar, adenosine, guanine with the sugar, guanosine, but hypoxanthine with the sugar, it's actually inosine, sir. So please take care about that. It's inosine. Perfect, done. That's the importance of minor purines. And ask to our students don't know about the minor purines at all. Minor purines पढ़ाते ही नहीं. In the MBBS days they will be just mugging up adenine guanine. Ask them minor purine, नहीं पता है. So that is the role of minor purine, sir. Hypoxanthine with the sugar with phosphate will be inosine monophosphate. And that is going to give rise to both of our purines here right now, sir. One is your AMP, another one is your GMP, sir. Okay. So in de novo synthesis, how to learn, guys? What is the starting substance? Ribose 5-phosphate. That will undergo certain reactions to form IMP, and IMP is going to form both your AMP as well as GMP. Khatam, done. Guys, can anyone tell me what is the meaning of de novo? De novo का मतलब क्या होता है? Let's go a little bit more in depth. Can anyone tell me what's the meaning of de novo? Come on. De novo. De novo is actually, the meaning of de novo is something new. Yes, very good. Something from the scratch, something from the very beginning. So listen here, what is the meaning? Why do doctors have given the name of this pathway as de novo synthesis here? Hello sir. This is the last and final point in the planning. After that we'll start with the reactions and all. और वो 10 मिनट में खत्म हो जाएगा सर. टॉपिक विल बी डन. So listen carefully, ये बहुत important है. My dear doctors, please look at this ribose 5-phosphate. Look at the ribose 5-phosphate and try to understand the meaning of that. What is the meaning of ribose 5-phosphate? Ribose 5-phosphate means that there is a sugar attached. अच्छा. Okay. That means if you are observing properly, मेरी नज़र से देखो. Okay. Look at the screen with my perspective in English. Look at the screen with my perspective here right now, guys. This will be the sugar molecule here, and then there is a phosphate group there. Oh my god, sugar bhi hai, phosphate group bhi hai. Already I'm having sugar there, already I'm having phosphate group there. Then what is going to happen right now? Come on everyone, listen here. Listen with all your ears open. Now comes glutamine. it will contribute nitrogen. Now comes tetrahydrofolate, carbon, aspartate, nitrogen, glycine, something like that, one by one all the substances will form one ring, and they are going to form one more ring. Do you remember these rings? Do you remember these rings? Tell me yes or no. Come on बच्चों, हाँ या ना बुलाओ, हाँ या ना, yes. तो all these substances will come one by one, बारी बारी से आते हैं, and they are going to contribute the carbon and nitrogen, and literally double ring structure will be formed here. And my dear friends, if this double ring structure is adenine, come on, adenine with the sugar, adenosine with phosphate, adenosine monophosphate, lovely, AMP. If agar मान लो कि this double ring structure is guanine, then guanine with the sugar, guanosine monophosphate, GMP, done. If that double ring structure is hypoxanthine, and then nothing but inosine monophosphate. Oh my god, that's the reason why doctors kept the name as de novo synthesis. Learn up to the research level. ऐसा नाम क्यों रखा गया है? रट्टा मत मारो फालतू में ना, de novo ऐसा क्यों रखा गया है? Why? Because that double ring structure is literally forming from the scratch, newly forming, carbon by carbon, nitrogen by nitrogen. That is why it is known as de novo synthesis of purines. Understanding here right now? And the platform is already ready in the form of ribose 5-phosphate. Sugar, ribose and 5-phosphate, ये बिल्कुल ready है. Sugar और phosphate ready है. Just add the double ring structure there, sir. Are you understanding my point here right now, guys? Hello. Okay, are you understanding everyone? Okay, so that is about, हाँ जी, ऐसे पढ़ना चाहिए, इन डेप्थ पढ़ना चाहिए तो मज़ा आएगा. अगर आपने ये समझ गया ना भाई, 80% टॉपिक डन. उसके बाद में सिर्फ 2 रिएक्शन हमको पढ़ना है, क्लास डन. ओके, तो इट्स ऑल अबाउट लर्निंग द ओवरव्यू से. चलो, come on, revise yourself. I will also be shouting from here, you will also shout from there, sir. Okay ना? तो sit at your homes and start shouting. Your parents will go mad. बंदा कौन पढ़ा रहा है, ये लोग क्या पढ़ रहे हैं? Synthesis of purines will be of how many categories? 2 categories. De novo synthesis, salvage pathway. De novo synthesis, what is the starting substance? Ribose 5-phosphate. Undergoes certain reaction to form IMP. IMP is going to form AMP and GMP. Class. De novo from the scratch. Okay, na? From the very beginning. And exactly opposite of that, salvage pathway का मतलब क्या होता है sir? Salvage pathway, I am not at all going to form the double ring structure. ये rings में form नहीं करूँगा. I will bring ready-made adenine, ready-made guanine, ready-made hypoxanthine, bring attach, salvage pathway, class. ठीक है जी? And this completes the complete overview. चलिए सर start करते हैं the first one, de novo synthesis, and then afterwards we'll study salvage pathway. अब यहाँ से fast part लिखते जाओ. Please write on the heading as de novo synthesis. Did you all understand the overview, guys? Gracie, समझ में आया तुमको? Now, what is the starting substance in the ribose 5-phosphate? Sorry, what is the starting substance here, sir? Ribose 5-phosphate. Okay. That ribose 5-phosphate is now going to get converted to phosphoribosyl pyrophosphate. Repeat this name as many times as possible in your mind, sir. Hundreds and thousands of times. Phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. क्योंकि ये बहुत important है. And in short, this is known as PRPP. बड़े प्यार से हम इसको ना PRPP कहते हैं, phosphoribosyl pyrophosphate. And what is the name of the enzyme here? The name of the enzyme, your favorite one, PRPP synthase. And in this reaction, we are going to use one ATP, and after using one ATP, it is getting converted to AMP, adenosine monophosphate. Reaction number 2. Reaction number 2. It is going to get converted to the phosphoribosyl amine. Phosphoribosyl amine. And suddenly, where are you getting that amine group from? Glutamine. glutamine will come here and it will contribute the amine group, and because the amine group is getting transported or transferred, therefore what is the name of the enzyme? Amidotransferase. What is the name of the enzyme, sir? Amidotransferase. After this nothing to be learned, sir. इसके बाद कुछ नहीं सीखना है आपको. Just do one thing here. Yes, mark few arrow marks here right now, sir. Write down there as IMP, and IMP is the one which is going to form your AMP, and it is going to form the GMP. Okay, sir. That's all. I promised you that there are only 2 reactions to be learned. Done, done, done. Are you all done? Very good, sir. So chaliye sir, you also tell the names, and I'll also be telling the names from here. Come on, my dear friends, tell me what is the starting substance? Ribose 5-phosphate that is going to get converted to phosphoribosyl pyrophosphate, PRPP, and that is getting converted to phosphoribosyl amine, and then it is going to form IMP. IMP is going to form AMP and GMP. Okay, चलिए इसको ना और आसान बनाते हैं. Everyone here right now, sir, and now comes the, this is like the textbook one. अब मैं आपको बताऊंगा, टेक्स्ट बुक और आपके दरमियान में मैं आता हूँ. I'll come in the middle of you and your textbook, है ना, to make it easy. Now let us see my part, sir, my version, how to make it easy. Ribose 5-phosphate, sugar, and there'll be a phosphate group on carbon number 5. Okay, ribose 5-phosphate. Okay, no problem. In this reaction, I'm using ATP. I request everyone to look at the screen. I'll not be able to repeat this again. मैं repeat नहीं कर पाऊंगा दोबारा. Okay. In this reaction, I'm using ATP. What is the full form of ATP? Adenosine triphosphate. There will be 3 phosphate group. From this, remove 2 phosphate groups. Sir. Whenever you remove 2 phosphate group together, it is known as pyrophosphate. I am telling again, whenever you remove 2 phosphate groups together, it is known as pyrophosphate. And now, my dear friends, bring that pyrophosphate from here. यहाँ से pyrophosphate लेकर आओ और यहाँ कार्बन नंबर 1 पर लगा दो pyrophosphate. Now please look at my screen here right now. Magic. This one is P R P P. What is that? Phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. PRPP. Phosphoribosyl pyrophosphate. Is it okay? अब हम क्या करेंगे पता है? ध्यान से देखो. अगर हम 2 pyrophosphate निकालते हैं यहाँ से तो क्या बचेगा? Only 1 phosphate. Therefore, that is why what is the name of this one? Adenosine monophosphate. अच्छा. Adenosine triphosphate becomes adenosine monophosphate if you remove the 2 phosphate groups from there. And now comes your glutamine, and the glutamine will give the amine group. क्या किया जाए? Remove the pyrophosphate, and after removing that pyrophosphate, what is left out? We are left out with phosphoribosyl, और यहाँ पर amine group लगा दो. Glutamine is coming and giving the amine group. बोलो बच्चों क्या बन जाएगा? phosphoribosyl amine. क्या बात है. Wow. Are you understanding now? अब कुछ तो बोलो. अब समझ में आ रहा है बच्चों? Very good. Thank you दिव्या. Thank you चौधरी. ओ चौधरी जी भी है. चलिए जी बढ़िया. यहाँ देखो तो ribose 5-phosphate is getting converted to PRPP, and PRPP is getting converted to PR amine. ठीक है. I think those 2 reactions are also made easy. Is it okay? And now my dear friends, And now, my dear friends, I don't know whether you are observing properly or not. If this is amine group, hello, think. If this is amine group, definitely what is the formula of amine? Amine formula will be NH2. If amine formula is NH2, oh, hydrogen को मारो गोली, नाइट्रोजन. अच्छा. Glutamine आ चुका है. Glutamine has come here and it has already given the amine group. Amine group formula is NH2, and in that NH2, forget about hydrogen, N, and it has given nitrogen there. That means ring formation started. ग्लूटामिन आ चुका है, अपना नाइट्रोजन दे चुका है, तो यहाँ से रिंग फॉर्मेशन स्टार्ट हो चुका है. अंडरस्टैंडिंग माय डियर फ्रेंड्स? दैट्स द ब्यूटी ऑफ द एंटायर स्टोरी. तो अब क्या किया जाए? रिमेनिंग आफ्टर दैट रिएक्शंस डोंट हैव टू लर्न. व्हाई? बिकॉज ईच ऑफ देम विल कम वन बाय वन एंड गिव द कार्बन एंड नाइट्रोजन. एंड माय स्टूडेंट्स आर ऑलरेडी एक्सपर्ट. तो लिखिए, नाइट्रोजन नंबर 1 इज गिवन बाय, कम ऑन, हु विल कम एंड गिव नाइट्रोजन नंबर 1? Aspartate. Who will come and give the nitrogen number 1? It will be aspartate. Okay, and after that carbon number 2 and carbon number 8 are given by tetrahydrofolate. Nitrogen number 3, nitrogen number हम कार्बन से जाते हैं ना. Yes, nitrogen number 3 and nitrogen number 9, these two will be given by the glutamine. ठीक है, glutamine. And then carbon number 4, carbon number 5 and nitrogen number 7, these three are given by glycine. And finally the carbon number 6 is given by carbon dioxide. If all these are given, double ring structure will be formed. It will lead to the formation of GMP sir. Class. One of the toughest topic in biochemistry. One of the toughest topic in biochemistry made easy, sir. I challenge, it can't be more easier than this one. So it's like done, sir. Thank you. So we are done with the first pathway, guys. De novo synthesis, done, sir. De novo synthesis, ribose 5-phosphate, PRPP, PR amine, and then IMP to AMP and GMP. So ho gaya, sir, done. And now welcome here right now, my dear friends. Once you are done with the de novo synthesis, the other side we are having the salvage pathway. Let's get started with the salvage pathway now. चलो सर, डिफिकल्ट वाला टॉपिक हो गया है, अब करते हैं टाइम पास. सैल्वेज पाथवे में हम क्या करेंगे? वी आर गोइंग टू ब्रिंग द रेडीमेड एडिनाइन, रेडीमेड गुआनाइन एंड रेडीमेड हाइपोजैंथीन एंड एटाइन. तो इसमें तो और भी टेंशन है सर. ओनली 3 पॉइंट्स, इट विल बी डन एंड दैट इट्स लाइक कॉपी पेस्ट. मेरे प्यारे प्यारे बच्चों, यहाँ देखो, दिस वन विल बी योर PRPP, phosphoribosyl pyrophosphate. And now bring ready-made adenine. Okay. 1 second. I request all of you to please stop writing and look at the screen. This is the only one thing to understand. From this PRPP, we are going to remove PP. हम यहाँ से ना PP निकाल देंगे. Once you remove pyrophosphate, I am writing that here. Okay, I have removed the pyrophosphate. तो बोलो बच्चों क्या बचेगा? What is remaining? I am remaining with, yes, the phosphate group here and the sugar here. And now transfer adenine. If you transfer adenine here, my dear friends, adenine with the sugar, ribose sugar, adenosine, and adenosine with 1 phosphate, adenosine monophosphate. मेरी नज़र से देखो, पहला प्यूरिन फॉर्म हो चुका है. ऐसा लग रहा है कि टाइम पास हो रहा है. इसको इंग्लिश में सैल्वेज पाथवे कहा जाता है. ठीक है जी. And now what is the name of the enzyme? On one side I'm having adenine. Okay. On the other side I'm having phosphoribosyl. Phosphoribosyl. And what I'm doing? I'm transferring. What is the name of the enzyme, sir? Transferase. Oh हो, ये तो और भी खतरनाक. क्या बात है सर. Adenine, and here I'm having phosphoribosyl pyrophosphate, but we have removed the pyrophosphate. So what is remaining? Pyrophosphate. Adenine and phosphoribosyl transferase. Done. अब यहाँ से सर enjoy your life, do copy paste. Phosphoribosyl pyrophosphate, PRPP. This reacts with the ready-made hypoxanthine. Remove this PP, and once you remove that PP, what is left out? Phosphoribosyl. Transfer hypoxanthine. You all know very well hypoxanthine with the sugar will be inosine monophosphate, IMP. And then again the same thing here, sir. Phosphoribosyl pyrophosphate, PRPP. React this with guanine. And when this reacts with guanine, again the same thing. Remove the PP. If you remove this PP from here, we are left out with phosphoribosyl. Transfer guanine. That leads to the formation of guanosine monophosphate. And I am done with the formation of AMP, IMP and GMP. Salvage pathway class. बोलो भरत, क्या बोल रहे हो सर? आज फाइनली डीएनए समझ आ गया. थैंक यू भाई. अरे यार आईपैड भाई साहब हो गया. Now listen here right now guys. Okay, now welcome here right now. Now what is the name of the enzyme for the reaction number 2 and 3, sir? In the reaction number 2, what is the name of the enzyme? It's hypoxanthine getting transferred here, and it is guanine getting transferred here. And for these two reactions we have got the same enzyme. Kaise? How? Look here. Hypoxanthine and then G for guanine. Hypoxanthine and G for guanine, and it is transferred on phosphoribosyl. Okay, welcome dear. Phosphoribosyl. Okay, now so hypoxanthine and then guanine, it is phosphoribosyl, and then transferase, HGPRT. Hypoxanthine guanine phosphoribosyl transferase, HGPRT. So इसका मतलब क्या है? What is the meaning of this one guys? The meaning of this one will be कि ना यहाँ पर ना दोनों के लिए separate enzyme नहीं है. We don't have a separate enzyme for both of them. कि hypoxanthine transfer, guanine transfer. We are having only one enzyme only, and that one enzyme will be hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine transferring on phosphoribosyl, HGPRT. And now we are done with the synthesis of purines, sir. If you remember, synthesis of purines will be done by how many methods, sir? 2 methods. De novo synthesis हो गया और salvage pathway हो गया, in the easiest manner possible. Done. And now we are going to learn the clinical correlation right now in some In some unfortunate people, yes, this enzyme will be deficient. This enzyme will be deficient. Okay. So therefore, write down the heading there as clinical correlation. The name of the disease will be Lesch-Nyhan syndrome. Underline this, put a star mark, highlight. मतलब जो करना है करो, घिस डालो. Extremely important nowadays, sir. Okay, in your MC exam, they're asking a lot of questions from the Lesch-Nyhan syndrome, sir. और मैंने आपको क्या सिखाया था बायोकेमिस्ट्री में? इन बायोकेमिस्ट्री ऑलवेज यू हैव टू लर्न व्हाट सर? एंजाइम डेफिशिएंट. एंड व्हाट इज द एंजाइम डेफिशिएंट हियर सर? HGPRT. Hypoxanthine, guanine, phosphoribosyl transferase. Okay, hypoxanthine, okay, guanine and phosphoribosyl transferase. iPad, okay. Yeah, देखो सर. So H for hypoxanthine and G for guanine and then phosphoribosyl transferase. Are you comfortable with this one here right now? And now what will be the clinical feature here right now, guys? Clinical feature इसका क्या होगा यहाँ पर? In these people are actually going to be having, you know, some mental disorders, you know, neurological disorders we can say. So better write down there as neurological disorders. These people are suffering with some neurological disorders. Okay, no problem. And number two, these people are also will suffering with the gout also. Okay, no problem. Okay, gout also. And then the most important thing is self-mutilation. And this one will be the keyword. ये ना keyword है सर हमारे लिए. ये important word है. Exams में the moment you see self-mutilation, just close your eyes and mark the answer as Lesch-Nyhan syndrome. And what is the meaning of self-mutilation, sir? Self-mutilation का मतलब क्या होता है? Mutilating मतलब harming one own self, our own self. अपने आप को harming को क्या कहते हैं हिंदी में? नुकसान पहुंचाना, ठीक है ना? Harming one own self, ठीक है ना? तो how these people will be harming their own selves, guys? Okay, कैसे अपने आप को harm करते हैं लोग ना? They'll be literally chewing their fingers. I'm not telling के biting nails and all. Nail biting नहीं कह रहा हूँ मैं. I'm telling literally chewing the fingers को chewing. तो all this will be damaged and it will be bleeding. They'll be literally chewing their own lips. अपने lips को ना खुद chew करते रहते. तो this will be all damaged and it will be bleeding. Understanding? So this kind of patients will be seen suffering with Lesch-Nyhan syndrome. Or the interesting part was there was an image based question. अब तुम लोग मेरे को गाली दोगे. There was an image based question in biochemistry. And the image based question was, there was an image given of the patient. I think you are able to see my video like an image only, sir. तो मान लो कैसा ना image दिया गया था और image में ना बंदे के ऐसे हाथ थे और वो सारे damaged थे. All these are damaged and it was bleeding, and here it was damaged and bleeding. And the question was, listen the question, interesting. In the given image, the enzyme deficient is, क्या बात है? In the given image, what is the enzyme deficient? The given image is self-mutilation, and in self-mutilation, what is the disease actually? Lesch-Nyhan syndrome. Okay, and in Lesch-Nyhan syndrome, what is the enzyme deficient? It is HGPRT. All the best. Okay, बच्चों. All the best. Now once you are done with this one here right now, guys, I am done with the synthesis of purines, and now let us learn about the degradation of purine, sir. चलिए अब हम पढ़ेंगे degradation of purine. We will do this in 2 minutes and then we'll take a break. Degradation of purine, sir. So my dear friends, what are purines? Purines are AMP and then GMP and then IMP. Adenosine monophosphate, guanosine monophosphate and inosine monophosphate. And all these three purines will undergo degradation, and after undergoing degradation, finally they are going to form hypoxanthine. Hypoxanthine is going to become what, sir? Xanthine. And xanthine is going to become what, sir? Uric acid. That's all, sir. And always the same question in your exams. Exam में वही एक घसा पीटा सवाल what is the end product of degradation of purine? What is the end product of degradation of purine? यूरिक एसिड. हमेशा दिल आज द सेम क्वेश्चन, और हमारे स्टूडेंट्स की जय हो कि हमेशा दिल बी मार्किंग यूरिया इन द एग्जाम एंड कमिंग बैक. भाई साहब, मैं हाथ जोड़ के आपसे कहता हूँ कि यूरिया और यूरिक एसिड अलग अलग है. यूरिया और यूरिक एसिड अलग अलग है भाई साहब. यूरिया और यूरिक एसिड में पता नहीं वाई स्टूडेंट्स आर गेटिंग कंफ्यूज्ड. एंड प्रोडक्ट ऑफ ब्रेकडाउन ऑफ प्यूरिन डिग्रेडेशन, uric acid. End product of degradation of purine is, okay, that is uric acid. And here you have to remember only one enzyme here, sir. The name of the enzyme here will be the xanthine oxidase. And here also we have got the same enzyme, that is xanthine oxidase. Okay. So AMP, GMP, all of them are actually undergoing degradation to form what's a uric acid, and uric acid is the end product of degradation. Okay. Now once this is done here right now, sir, in some patients what is going to happen, the uric acid level will increase, and that condition is known as gout. So please write down clinical correlation. Gout. And what is gout, sir? Gout is nothing but hyperuricemia. Hyper means increased uric acid, emia means blood. So in the blood there will be increased level of uric acid. Uric acid level क्या होता है? बढ़ जाता है. ठीक है जी. And because of that increased level of uric acid, it is going to form urate crystals, sir, and that it is going to form the monosodium urate crystal. Monosodium urate crystals, sir. I mean to say MSU crystals. Is it okay? And again the same thing right from first class to last class. I told in the very first class ic acids will form 8 salts. यहाँ पर भी वही है, uric acid will form urate crystal, urate salts, sir. Okay. And there is a condition also known as pseudo gout, sir. And that was the question in your exam. I will also give you that like a homework only. In pseudo gout, find out homework which crystals. In gout, monosodium urate crystals are deposited. In pseudo gout, which crystals are deposited? Find out, sir. Okay, na? And that completes this topic of nucleotide metabolism, sir. Nucleotide metabolism topic, class done. Synthesis of purine and then degradation of purine, sir. ठीक है जी, हो गया? Yes, iPads. That uric acid levels, urate crystals will be deposited in joints. That is why it will lead to gouty arthritis. Arthritis होता है, ठीक है ना? सर इसमें एंजाइम डेफिशिएंसी नहीं है, इंक्रीज अमाउंट्स ऑफ यूरिक एसिड है सर, एंजाइम डेफिशिएंसी नहीं होता है, ठीक है ना? गुड. ओके, सो दैट कंप्लीट दिस टॉपिक हियर राइट नाउ, सर. तो आप लोग ना आराम से बैठ के 2 टॉपिक रिवाइज करो, एंड विल टेक ए स्मॉल ब्रेक हियर फॉर 10 मिनट्स. एंड प्लीज टुडे नॉट मोर देन 10 मिनट्स, सर. 10 मिनट से ज़्यादा नहीं. Don't waste your time today. So exactly the break will be for 10 minutes, and I'm going. Let's get started right now, guys. Okay, and now we're going to begin with and now we are going to begin with the molecular biology right now, guys. ठीक है? Let's begin with the molecular biology. Please write on the heading there as molecular biology. Yes. Molecular biology, sir. And this molecular biology, it is actually going to take exactly 2 hours of time. So let us just complete this molecular biology and finish it off, guys. And I want like complete concentration here right now, guys. Okay, everyone here. So this molecular biology part, And now guys, the molecular biology part, we are going to first of all I'll give you the overview of the molecular biology part here right now guys. Okay, and now suppose if this to be your cell here right now. So okay, I'm just giving you the overview. Listen here carefully guys. The planning, what we are going to learn for the next 2 hours. Suppose if there is a cell, and inside the cell मान लो के suppose this one is a nucleus here right now guys. ठीक है, and inside this nucleus what is going to happen, you will have DNA, conversion of a DNA to a DNA, I mean to say formation of a copy of DNA from a DNA. This process is known as replication, and you don't even have to mug up right now guys. We are going to form a copy of DNA. In some good English we are forming the replica of the DNA. So a replica of the DNA, a replication. Now DNA to RNA, this process is known as transcription. Transcription. So DNA to DNA will be replication, and DNA to RNA will be transcription. Definition done. Not even definition, even location is also done. You can see my diagram and tell me okay, it is taking place in the nucleus. And now what is going to happen, if this is like messenger RNA, if it is like mRNA, the messenger RNA will come out into cytoplasm, and in the cytoplasm mRNA helps in the formation of protein, and this process is known as translation. That's all. So this is what we are going to learn in the next 2 hours. 2 घंटे में पढ़ने वाले हैं सर, ठीक है. So my dear friends, चलिए एक बार रिपीट करते हैं. What is the definition of replication guys? DNA to DNA, and where it is taking place, nucleus. What is, and it is taking place in nucleus. And what is the meaning of translation? Translation means RNA to protein, and that is taking place in cytoplasm. And here itself I want to even tell you about the enzyme also. Remember, the enzyme which will be doing replication. Replication is done by DNA replication is done by an enzyme known as DNA polymerase, whereas transcription is done by an enzyme that is known as RNA polymerase. Okay, and we don't have like specific enzyme for translation guys, so don't worry about that. So the enzyme that is required for replication process is DNA polymerase, and enzyme for transcription will be RNA polymerase guys. Okay, so that is the entire picture, and out of this first let's learn about the replication, and then afterwards we'll learn about the transcription and translation. So my dear friends, please write on the heading as replication. Now the definition of the replication you people already know. What is replication guys? We are going to form a copy of DNA from a DNA. We are going to form a copy of the DNA from a DNA. That is nothing but replication. And you also know what is the name of the enzyme here. The name of the enzyme here is DNA polymerase. But you know, please look at the screen here. We are actually going to form this daughter DNA. And to form that daughter DNA, we have to depend on a parent DNA. We have to depend on this parent DNA. So because we are depending on a DNA, the complete name of the enzyme is DNA dependent DNA polymerase. I repeat again, because you have to depend on this DNA, that is why the name of the enzyme will be DNA dependent DNA polymerase. DNA dependent DNA polymerase. But during studies, we are not at all going to learn the full name of the enzyme. We are just going to learn only the partial name or the small name, that is DNA polymerase. That's all. We are not going to learn like DNA dependent DNA polymerase, only DNA polymerase. Next, definition done, enzyme name done. After that, the location. Where is replication taking place? Yes, we all know it is taking place inside the nucleus. Replication is taking place inside the nucleus. Okay, and now my dear friends, the good news is that if we and all सब कुछ हो चुका है biochemistry में, all the reactions are done. Now it's only about beautiful stories. Okay, so it's like replication is like one kahani. Similarly, transcription, translation is like one kahani. ठीक है ना? So remember, it's like one story. Are you getting a point here right now? So therefore, let's begin the story. It will take like 15-20 minutes to complete the entire replication process. I will tell you आराम से slowly learn it properly guys. Is it okay? तो चलो सर start करते हैं. In replication, my dear friends, first of all, you'll be having a double-stranded DNA. And first thing, what is going to happen, sir? It should be, both the strands should be separated. Sir, I'm telling you, I'm going to tell you slowly and please try to understand at once. Complete concentration here means here. सबसे पहले हम क्या करेंगे, डबल स्ट्रैंडेड को दोनों स्ट्रैंड्स को हम सेपरेट करेंगे. सो वी आर गोइंग टू सेपरेट बोथ द स्ट्रैंड्स ऑफ डीएनए. सो दिस इज लाइक वन स्ट्रैंड ऑफ द डीएनए. Okay, and this will be like another strand of the DNA. And the first thing what is going to happen here is that yes, both the strands of DNA are separated apart. And who is going to separate the DNA strands? It's DNA A protein. So DNA A protein is going to separate both the strands of DNA. So DNA A protein will separate both the strands of DNA. DNA A protein separates both the strands of DNA. Done. Now once you are done with the separation of this one guys, so what you have to remember is what is the name of the substance and what is the function of the substance. So who is helping in separating the strands? DNA A protein. But now the problem, find the problem, solve the problem. Who is going to separate the strand? DNA A protein. But after separation they will not be separated, they will again try to recoil back. वापस वो फिर से recoil होने की कोशिश करते हैं. तो it's not enough that you only separate them, you need to even keep them separated. उसको ना separate भी रखना पड़ता है. That is known as stabilizing them. And these are stabilized by SSBs. SSBs. And the name is very meaningful. SSB actually stands for, yes, single strand binding protein. So this will go and bind to the single strand and they will stabilize. Stabilize का मतलब क्या होता है? What is the meaning of stabilize? They will prevent that from coiling. Coiling से वो रोकते हैं. तो चलिए सर, finally we have opened that. So I repeat again, who is helping in separating both the strands? DNA A protein. And who is keeping them separated apart, stabilizing them, and stabilizing them, that is done by SSBs. Now welcome to the third one sir. दो चीजें हो चुकी है. Welcome to the third thing right now. Now my dear friends, what is going to happen is this strand of the DNA is going to form, you know, its own new strand, and this strand of the DNA is going to form its own new strand. So both the strands of the DNA, they are going to form their own new strand. Okay, but tell me, will the replication occur only in this much part of the DNA? Will replication occur only in that much part? No, entire DNA has to undergo replication. Entire DNA has to undergo replication. क्योंकि यार नाम में लिखा है, इसके लिए ना कोई आपको difficult science नहीं है. You don't have anything like very difficult concept. The name itself is telling you replication, and replication means what? There is a formation of replica. Replica का मतलब क्या होता है? Formation of copy, exact copy. And for the forming exact copy of the DNA, entire DNA has to undergo replication. So therefore, now what is going to happen, guys? This helix has to be uncoiled. This helix has to be uncoiled, and it is done by the third enzyme, that is helicases. It is done by helicases. Helicases क्या करते हैं? उसको ना uncoil करते जाते हैं. They'll keep on uncoiling that. Helicases keeps on uncoiling that. Number 3. I repeat again, who is separating the strand? DNA A protein. Who will stabilize that? SSBs. And who is uncoiling the helicases? Now the last and final one, so the fourth one right now. After that the story changes. उसके बाद में कहानी change होती है और interesting हो जाएगी कहानी. ध्यान से देखो. Now the fourth thing you need to understand with an example. For example, I'm taking two ropes. I take two ropes, and these two ropes I'm going to coil them each other. Coil them, make a coil. And after making a coil like a DNA, there are two ropes. I'm trying to separate them apart. आपने ना ये एक्सपेरिमेंट किया होगा विद द थ्रेड्स एंड ऑल. हम लोग कभी कभी ना धागे के साथ में एक्सपेरिमेंट करते हैं ना, दो धागे लेके ऐसे ना कॉइल करते हैं. जब उसको सेपरेट करोगे, व्हेन यू आर ट्राइंग टू सेपरेट द टू ropes, the two threads, when you're trying to separate, I think you have done this experiment, you will be able to separate that up to certain extent, but beyond that they will not be separated. Beyond that the coiling is going to become more tighter. अंडरस्टैंडिंग. So those two ropes, they are separated up to certain extent, लेकिन उससे आगे क्या होगा, they are going to become more tighter. Exactly the same thing is happening in the DNA also. Helicases are trying to uncoil, but what is going to happen, they are able to uncoil up to certain extent, but beyond that the coiling is becoming like more tighter, और इसको हम कहते हैं super coil. Super coil. अब यार क्या होगा, if the super coil is formed, then what is going to happen, further it is unable to uncoil. If further it is unable to uncoil, hello, if further it is unable to uncoil, can we stop in the middle? No. Why? Because you only told me entire DNA has to be replicated. Complete DNA replicate होना चाहिए. तो चलिए सर इसको हम कैसे uncoil करते हैं, यहाँ पर देखिए. Now welcome the fourth player, that is topoisomerase. And topoisomerase, what they are going to do is that they are going to cut one of the strand of the DNA. And after cutting one of the strand of the DNA, they are going to uncoil, relax the supercoil, and then again join them together, ligate them together. इनके दो काम होते हैं. Strand को cut करेंगे, they are going to make a nick and then relax them, uncoil them, and then again seal them together. Nick and sealing कहते हैं उसको, ठीक है ना? That is done by topoisomerases. Is it okay, my dear friends? So therefore you have to remember about these four things here right now. DNA A protein, I repeat for the last and final time, DNA A protein separating the strand, SSBs stabilizing, helicases uncoiling the helix, and topoisomerase will relax supercoil. And please write down some more detail about topoisomerase below the diagram. Topoisomerase, what is the function of this one, sir? Yes, it will be helping in nick and sealing. Nick and sealing, and by this what it is doing, sir, it relaxes supercoils. Nick and sealing, it is relaxing supercoils. And guys, yes, topoisomerase will be again of two types, sir. Topoisomerase 1 and topoisomerase 2. And I want you to remember only one point here. Topoisomerase 2 is also known as DNA gyrase. This enzyme is important, it will come in pharmacology. There are certain drugs inhibiting that one, sir, DNA gyrase. And DNA gyrase is nothing but another name of topoisomerase 2, sir. Are you comfortable? Are you comfortable? Is it okay? So topoisomerases will be of two types, sir. That is topoisomerase 1 and topoisomerase 2. And topoisomerase 2 is also known as DNA gyrase. That's all done. So by this we complete the introduction of replication. अब सुनो ध्यान से सर, यहाँ से स्टार्ट होती है एक्चुअल कहानी. Now my dear friends, hello. I am done with all the four of them here, sir. Who is separating DNA A protein, who is stabilizing SSBs, who is uncoiling the helicases, and then finally who is relaxing the supercoil, that is topoisomerase. ठीक है, हो गया. And now, my dear friends, tell me, did we start with replication till now? क्या हमने replication process start किया है? Did we start with replication process now, till now? Come on. We did not start with replication process. Perfect. How can you say that? Just few minutes back I told you replication is done by which enzyme? DNA polymerase. And now tell me, my dear friends, did DNA polymerase come here till now? Did DNA polymerase enzyme come here till now? No. And when there is no DNA polymerase enzyme here till now, there is no replication started till now. So from that time, what are we doing? From that time, we are actually making the platform ready. Okay. So our DNA polymerase enzyme, from today onwards, remember the story, like, DNA polymerase enzyme is like a VIP. And before the VIP comes, arrangement has to be made. So DNA polymerase enzyme is like, I am not going to come there and separate the strand. I'm not going to stabilize. I'm not going to uncoil helix. I'm not at all going to relax supercoil. Sub ready rakho. Do everything, keep it ready. I'll just come, replicate and go away. Wow. And to make everything ready, we have employed 4 people here. Who are the four people here? DNA, A protein, and then helicases, SSB, and topoisomerase. And they have kept everything ready. Now let us welcome DNA polymerase. So please come and replicate. Understanding here right now? So my dear friends, that is how just make a story. So these four people are keeping everything ready. Now let's welcome DNA polymerase. So do one thing, guys. Take a fresh page in your books and please make your book horizontal. Please take a fresh page in your book and please make your book horizontal. Landscape mode. Imagine this is like your horizontal page and draw the diagram exactly as I am drawing, sir. Big, neat and clear. Last diagram, इसमें सब कुछ हो जाएगा, sir. Everything will be done in this diagram. Now imagine that this is like one strand of the DNA. That is from 3' to 5'. And imagine that this is another strand of the DNA. And this is anti-parallel, so here it will be 3' and here it will be 5'. And how it is anti-parallel and everything we have learned in the basics. That's why I told you basics are important. Now guys, this is like exactly the same diagram. This is like exactly the same diagram only. We have already separated the strand. The only thing is that, you know, we have removed everything with the names and everything. Now a bigger diagram. And now my dear friends, everything is ready. Everything is ready. And now please welcome the enzyme DNA polymerase. Now comes your enzyme DNA polymerase, hero of the story. This is our hero of the story, DNA polymerase. And once the DNA polymerase enzyme comes here, मैंने कहा ना आपको it's like a story, कहानी से, learn it like a story only. So once the DNA polymerase comes here near the DNA, now there is a problem. What is the problem? It's unable to sit on the single strand and do replication. So what to do for this one, sir? It is unstable. You have to make it stable. How to make it stable? To make it stable, we need to attach a small single strand. And that small single strand is known as primer. इसको हम कहते हैं primer. And because it is single strand, it is known as RNA primer. So there is a small single strand attached there, RNA primer. And because that single strand RNA primer is attached, it is just observe that carefully. It is looking like a double strand. So now comes your enzyme and sit here comfortably. So what is our first problem? Welcome DNA polymerase. After it is coming there, what is our first problem? It is unstable. Stabilize by attaching a primer. And now the question comes here, sir, who is forming this RNA primer? RNA primer कौन form कर रहा है? It is formed by an enzyme. The name of the enzyme is primase. Primase is an enzyme which is going to form RNA primer. RNA primer is formed by primase. Done. चलिए सर, तो we have stabilized. कितना मस्त कहानी है ना? सो डीएनए पॉलीमरेज आ चुका है और डीएनए पॉलीमरेज वहाँ पर तशरीफ रख चुका है, ओके ना, बैठ चुका है. एंड आफ्टर सिटिंग देयर कंफर्टेबली स्टेबल, नाउ इट विल स्टार्ट रेप्लिकेशन. इन विच डायरेक्शन? दैट इज इंपॉर्टेंट. ओके, नाउ डीएनए पॉलीमरेज हैज कम हियर एंड आफ्टर दैट स्टेबिलाइज द डीएनए पॉलीमरेज विद द हेल्प ऑफ अ स्मॉल सिंगल स्ट्रैंड. व्हाट इज द नेम ऑफ द सिंगल स्ट्रैंड? प्राइमर, आरएनए प्राइमर. एंड हु इज गोइंग टू फॉर्म दिस सिंगल स्ट्रैंड आरएनए प्राइमर? प्राइमेस. एंजाइम नेम प्राइमेस. ओके भरत. After sitting here, now it is going to start replication. In which direction? Remember, reading will be from 3 to 5. You can clearly see there is a 3' here and there is a 5' here. So reading will be always from where to where, sir? 3 to 5. Reading the strand. So it is going to read 3 to 5. How? Yes, today only we have learned in the very first topic. मान लो कि there is adenine, thymine, adenine, guanine, cytosine, guanine, adenine, something, sir. We already have learned in the very first topic today. DNA strand will be a polymer of nucleotide. So this enzyme is going to read that in which direction? 3 to 5. After reading in 3 to 5 direction, it is going to form the new strand. If it is adenine, it will attach thymine. If it is thymine, adenine, adenine, thymine, guanine, cytosine, guanine. Now, if I ask you a question, how can you say it is attaching like this? Of course, sir has taught us the basics. This is Chargav's rule. Chargav bhai sahab ne kaha tha, adenine loves to bind with thymine. So if it is adenine, thymine, thymine, adenine, something like that. In this manner, the new strand will be formed. Okay. Basics, basics, basics. Sir. If basics are clear, you will enjoy the concept. Now, if this new strand is formed here, now can anyone tell me the new strand is formed in which direction? Obviously this will be 5 dash and obviously this will be 3 dash. Sir, how can you tell this is like 5 dash and 3 dash? Why? Because again go back to the basics, first topic. Sir explained that a DNA will be having double strand and that double strand will be anti-parallel to each other. So therefore if this is like 3 to 5, if this parent strand, black colored line, is 3 to 5, obviously this will be 5 to 3. So the new strand will be always formed in which direction, sir? 5 dash to 3 dash. And now these 2 points should be like printed in your mind like 2 rules. Reading will be always 3 to 5 and formation of new strand will be 5 to 3. Is tarha se, in this manner, this enzyme will keep on reading and keep on forming the new strand, no problem. Now my concept is done. Now what you do is whatever I taught you here, just apply that concept onto the other strand right now, sir. दूसरे strand पर apply करिए, exactly copy paste. Come on. Start. Welcome DNA polymerase. Okay. After coming DNA polymerase is unstable. Okay. How to make it stable? Attach primer. Who is going to form the primer? Enzyme primase. After attaching the primer, the enzyme DNA polymerase will come and sit here. And it is going to read this strand. I'm not going to write again. After reading that, it is going to form the new strand. Reading will be in which direction, sir? Yes, 3 to 5. Apply my rules. Reading will be 3 to 5. And formation of the new strand will be obviously in 5 dash to 3 dash direction, in this direction. Okay. Wow. Are you all comfortable till here? Are you all comfortable till here? Perfect. So welcome DNA polymerase, and DNA polymerase enzyme will come and sit. Okay, welcome DNA polymerase. The DNA polymerase enzyme will come and sit. Okay, with the help of the primer. Primer will make it stable. With the help of the primer, it will come and sit there. Reading will be in 3 to 5, and the formation of new strand will be 5 dash to 3 dash. And now my dear friends, this is something confusing right now. अब क्या होगा? Will replication occur only in this much area? No. Replication will occur in the entire DNA. Entire DNA will undergo replication. So therefore what I am going to do is further open. And who is going to open or this helix, guys? Who is going to open this helix? Helicases. Helicases are going to uncoil the helix. And now my dear friends, something additional here which I did not tell you. Look here. After separating now, after separating here, what is happening, you know? 1 second, I'll tell you. Yes. Just observe here. After separating, this entire thing is appearing to me like a bubble. And that is why it is known as replication bubble. इसको हम replication bubble कहते हैं. And in this replication bubble, I am able to find one end here and one end here, and this is known as the replication fork कहते हैं. It is known as replication fork. I wanted it, did not tell you before. Now it is needed. So there will be one end here replication fork, and this one end here replication fork. And now my dear friends, welcome back to our diagram here right now. So this is a replication fork here, and there is a replication fork here. And listen this thing very carefully. 200% concentration here. Every student will be confused here only, sir. Please, you people don't get confused here right now. You need to study now either in this direction or in this direction. Just for the sake of studying. पढ़ने के लिए किसी एक direction में पढ़ना पड़ेगा. You need to study in one direction. And for example, we are studying with respect to this replication fork. What I am telling you, I know that there is one more replication fork here, but we are now studying with respect to this replication fork. Whatever. Either you can study on right side or left side, whatever it is. I am selecting this one. Come on. Now tell me, with respect to this replication fork, what about this strand, sir? Is it moving towards the replication fork or away? Tell me, is it moving towards or away? Is it moving like towards the replication fork or away from the replication fork, guys? Can you please answer me? Yes, it's moving towards. And remember, the strand which is moving towards the replication fork is known as the leading strand. The strand which moves towards the replication fork is known as the leading strand. The strand which moves towards the replication fork is known as leading strand. Leading strand. Is it okay? So therefore, this leading strand is moving towards the replication fork, and no problem with that. It will keep on reading, keep on forming, keep on reading, keep on forming. So no problem with that. Now the strand, the strand below here, you can clearly see it is going away from the replication fork. So because it is moving away, it is known as lagging strand. So what is the definition if someone asks you, sir, what is the meaning of leading strand? The strand which moves towards the replication fork is leading, and the strand which moves away from the replication fork will be lagging. And now here there is something really interesting concept here right now, guys. Come on. Sir, leading strand, yeah, ये read करता चला जाएगा, form करता चला जाएगा. It will keep on reading, keep on forming. No problem with the leading strand. Now the problem comes with the lagging strand. Lagging strand is going here, away from this replication fork. Then my question is, basic question. यहाँ पर replication होना चाहिए कि नहीं होना चाहिए? Tell me yes or no. Should replication occur in this region or not? Yes or no? Should replication occur in this region? Yes, no doubt about that. No doubt about that. Entire DNA has to undergo replication. Entire DNA has to undergo replication. Yes, यहाँ होना चाहिए. If replication has to occur here, there's a problem. What is the problem? It's going away. If it is moving away, then what will happen in this region? यहाँ पर replication कैसे होगा? How? Repeat the story. Repeat the story. Welcome DNA polymerase enzyme. After welcoming DNA polymerase, attach a primer. Listen carefully. After attaching primer, DNA polymerase enzyme will come and sit there. After sitting there, it is going to read in 3 to 5, forming the new strand in 5 to 3. It's forming. So it is going to form the new strand in 5 to 3 direction. 5 to 3 direction. Is it okay? It will go and stop there. And again the same question. Should replication occur in the remaining area? Yes. What to do? Repeat the story. Welcome DNA polymerase. Form the primer. DNA polymerase will come and sit there. Reading 3 to 5. Formation of new strand. Reading 3 to 5. Formation of new strand 5 to 3. Forming. It will come and stop here. So in this manner it is going to form, sir. So the leading strand is forming continuously and the lagging strand is forming discontinuously. Leading strand is actually forming towards replication fork, and remember it is a continuous strand. Lagging strand is forming away from replication fork, and this one will be discontinuous. And you can clearly see there, guys, this lagging strand. Hello everyone, the final conclusion why I'm doing all this hard work with you. This lagging strand which is discontinuous, it is formed in the form of small fragments, and these fragments, small fragments, are known as, oh my god, Okazaki fragments कहते हैं sir. I hope you have just mugged up this one, guys. Lagging strand will consist of fragments, and those are known as Okazaki fragments. हो गया? Is it okay? So they will ask the question in the exam, Okazaki fragments. Okay, yeah, Okazaki is a normal thing. Okazaki fragments, they will ask you the question, they are present in which strand? Yes, you can answer that. It is present in the lagging strand. Okazaki fragments are present in lagging strand. Very good, Gracie. So my dear friends, let me just repeat this once. The strand which moves towards the replication fork, leading, and the strand which is moving away from the replication fork, lagging. Which is a continuous strand, leading, which is a discontinuous strand, lagging strand. And which strand will be having Okazaki fragments, and that will be the lagging strand. Lagging strand has got the Okazaki fragments. That's all. हो गया. And मेरी नज़र से देखो यहाँ पर, just observe here, from this parent DNA we got like two daughter DNA. Like in this diagram we are able to see here, sir, double stranded, sir. And in that the black colored one old strand and one new strand, old strand and new strands. So I repeat again, from one parent DNA you got the two daughter DNA. I don't have any problem with this daughter DNA. That daughter DNA is perfect, but I am having a problem with this daughter DNA. Now what is the problem with this daughter DNA? Yes, the problem with this daughter DNA is the primer. So in the middle you are having RNA primer. Now tell me, my dear friends, in DNA structure, have you ever learned in your life कि वहाँ पर primers होते हैं? In DNA structure, do you have like primers? No, the primers will not be there. तो क्या किया जाए? What to do right now? Remove the primer. But after removing the primer, there is a problem. What is the problem? Gaps. So fill the gap, gap filling. After filling the gap, one more problem, they are not attached. Ligate. Now it's a perfect DNA, sir. Are you understanding your point here right now? So in this you have to remove the primer, fill the gap, and then ligate. And for that, let us write down 4 points and finish it also. So write down the 4 points regarding RNA primer. Okay, now RNA primer. Initially, in the very beginning of the topic, we have learned the formation of RNA primer. Who is going to form the RNA primer? RNA primer will be formed by primase. And at the end of the topic, we have learned about the removal of the primer. Who is going to remove the primer? The primer will be removed by RNase H. RNase H is going to remove the primer. And after that removal of primer, there is formation of gap filling. And who is now going to fill the gap? The gap filling is done by DNA polymerase. And which DNA polymerase, sir? Gap filling is done by DNA polymerase 1. What do you mean by that, sir? That means that, from that time, this DNA polymerase which is doing replication, actually it is DNA polymerase 3. Wantedly I am telling you now, please update that, sir, so that you can understand the difference. इसीलिए मैं आपको ना अभी बता रहा हूँ. So this is DNA polymerase 3 here right now, guys, which is doing replication process. Replication process is done by DNA polymerase 3. Leading strand, lagging strand, and whereas gap filling will be done by DNA polymerase 1. And after that, the last and final point that you have to remember, yes sir, ligation. Ligation of the strands is done by, yes, DNA ligase. Very good. And that completes your replication process. Oh yeah, it's like a nice story. समझ में आ रहा है? Are you getting a point here right now, guys? And that completes our first topic, replications. So who is going to form the primer in the beginning? Primase. Who is going to remove the primer? RNase H. And who is going to fill the gap? DNA polymerase 1. And who is going to ligate that? DNA ligase. Is it okay? So that completes the process of the formation of, DNA from a DNA. Copy of a DNA from a DNA. Now here, some students might be having a doubt in their mind. Hello. Some students might be having a doubt in the mind. Sir, you have explained on the right side here. I don't want to study here. I want to study on the left side. It is exactly the same. This will become leading, moving towards, and this will become lagging. Understanding your point here right now? So if you are studying with respect to this side here, I repeat for the last time, please don't ask me this again and again. If you study from this side here, this will become your leading and this is lagging. Moving towards leading, away, lagging. Replication fork. No sir, I don't want to study on this side, I want to study on this side. No problem. There also the concept is not going to change. This with respect to this one, इसके मुताबिक according to this replication fork, ये वाला बन जाएगा towards means leading और ये वाला बन जाएगा lagging. Understanding? So either you study or right or study or left, it will be exactly the same concept. Concept is not going to change. Is it okay? Done sir. That completes your entire replication process. Is it okay? I'll give you 2 minutes of time. Please revise this. Then we'll start with the protein synthesis, transcription and translation. Thank you, Chaudhary. Replication fork दोनों साइड में होती है. iPad 2, I said many times. Jayashri, interesting class sir. Thank you Jayashri. Thanks for appreciation. Arjun says it means actually both the DNA will not be discontinuous, Arjun. Continuous as well as discontinuous. Other discontinuous, other continuous. Other discontinuous, other continuous. It will be half continuous, half discontinuous. Gracie says crystal clear. Thank you sir. Welcome Gracie. Removal enzyme is RNase H. मैंने आपको कहा था ना in biochemistry whenever you are removing ACE, so it is RNA primer. RNA primer को remove करने के लिए RNase H. चलो, welcome everyone. Thank you. Asking about DNA, yeah, DNA polymerase 2 will also be there. It is required for repair of DNA. That is just memorizing part. You need to memorize that. Actually, we are having different types of enzymes and their function will be there. Like DNA polymerase 1, it is helping in gap filling. DNA polymerase 3 will be helping in replication. DNA polymerase 2 will be helping in repair. तो उसमें कुछ पढ़ाने लायक नहीं है. There is nothing to teach in that. You need to just memorize the name of the DNA enzyme and name of the function. Bharat says lovely. Thank you Bharat. Thank you all. चलो सर, अब हम बढ़ते हैं आगे. Now once you are done with this one right now guys, okay, iPad 2 says sir anatomy भी करवा दो. सर जी करवा दी anatomy. Raju, thank you. Now once you are done with this replication guys, I hope you people are like perfect with replication. And now the other side we are going to do the transcription and translation, and this is actually the protein synthesis. Okay, so write on the big heading protein synthesis. So in this way only we are going to do slowly, comfortably, aram se. Please write down the heading as protein synthesis. I think it will take another 40-45 minutes of time to do this process, and if this is done my dear friends, biochemistry is done. We are left out with a small topic, enzyme. Enzymes topic will be hardly done in 20-25 minutes, and that will complete biochemistry. So it's hardly a matter of another 2 hours. In another 2 hours biochemistry will be done. Okay, so please concentrate here right now guys. Okay, now protein synthesis. Let me again do the same thing. Let me first of all give you the overview. In protein synthesis, we are going to begin with DNA, and DNA is undergoing the process of transcription to form hnRNA. As I told you, I am going to go a little bit in more depth. This is heteronuclear RNA, and this process is known as transcription. DNA will undergo the process of transcription to form hnRNA, heteronuclear RNA. Heteronuclear RNA. And after forming this heteronuclear RNA, this will undergo certain modification to form your actual mRNA. Okay. So that is the reason why this is known as post-transcriptional. After transcription, you know, post-transcriptional modification. And this is extremely important topic for your MCI exam. I don't know why MCI fellow gives a question from that topic every time. Halaki, I'll teach you now. Okay, there is hardly like 2 minutes topic. So there are only 3 points in that, and from that 3 points only that waste fellow will keep on giving the question. Don't know why. So DNA to hnRNA transcription, and hnRNA will be modified. That is known as post-transcriptional modification. Where is this taking place? Of course, it is taking place inside the nucleus. And what is coming out from the nucleus into cytosol will be your mRNA. Now mRNA will come out into the cytosol. After coming out into the cytosol, mRNA, and then that will lead to the formation of protein form होता है. The post translational modification. So DNA to hnRNA, I repeat again, DNA to hnRNA, that is transcription, hnRNA to mRNA, that is post transcriptional modification, and then mRNA to protein will be translation, and the protein will be modified, and that is known as the post translational modification. Is it okay? Are you done? So basically what I'm trying to tell you here is that these are the 4 topics that you need to learn. These are the 4 topics that we have to learn here right now, sir. So let us see all these 4 topics one by one. First, let's start with the first topic right now, sir. Transcription. Let's start with the first topic right now, sir. Transcription. Again, DNA to RNA. So please be very careful, guys. DNA will be double stranded and RNA will be single stranded. So from a double stranded DNA, we are going to form single stranded RNA. I'm requesting you, all of you, I'm not ordering you guys, I'm requesting you, please 40 minutes, please concentrate here right now, because as the topics will keep on moving ahead, there'll be a lot of confusion and the topics will be mixing up, replication will come into transcription and all. So please, I'm requesting you, don't distract yourself. Transcription means double stranded DNA to single stranded RNA. It will be done in 5 minutes. Everyone look here. Please take a fresh page in your book and please make your book horizontal. Please take a fresh page in your book and please make your book horizontal. Okay, landscape mode, ऐसा horizontal करो. On the top of the page, write down there as transcription. The first topic will be done on one page in 5 minutes. Definition of transcription, you people are already perfect with that. That is nothing but DNA to RNA. And what is the name of the enzyme which is going to do transcription, guys? That is RNA polymerase. And for the formation of this RNA, you have to depend on DNA. That is why complete name will be DNA dependent RNA polymerase. Oh my God. For the formation of RNA, you are going to depend on the DNA. That is why it is known as DNA dependent RNA polymerase. DNA dependent RNA polymerase. And where is this transcription taking place? So what is the location of transcription? Of course, it is taking place inside the nucleus. Transcription is taking place inside the nucleus. Is it okay? Fine. So transcription means DNA to RNA. What is the name of the enzyme, sir? The name of the enzyme is DNA dependent RNA polymerase. And then location of that will be inside the nucleus. And now let's start with the process. Okay. Now from here onwards, it will be exactly 5 minutes, sir. So first of all, I am taking a DNA. One strand of the DNA will be 3' to 5', and the other strand of the DNA will be like 5' to 3'. Okay, imagine like this is like our double stranded DNA. मान लो कि ना ये वाला क्या है अपना DNA है सर. Now from this double stranded DNA we have to form what sir? RNA. Single stranded RNA form करना है. And remember it is taking place in how many stages? 3 stages. Initiation, number 2 elongation, and number 3 termination. So there is something known as 3 stages में होता है सर. Initiation, elongation, termination. And now first of all do one thing, decide कि will entire DNA form RNA or only a part of DNA will form RNA. So remember my dear friends, only and only a part of DNA is going to form RNA. Last topic that we have learned was replication. Replication in simple English means what? Replica. Replica means exact copy. So exact copy of the DNA will be formed in replication process. Replica, DNA to DNA. Replica, DNA to DNA. Understanding here? But in transcription, only a part of DNA will undergo transcription. Okay, so therefore in this process only a small part of the DNA is undergoing transcription. So you just imagine this part of the DNA is undergoing transcription. You just imagine some sequence: guanine, cytosine, cytosine, guanine, thymine, adenine, something, okay, something like this, whatever. Now our platform ready sir. Come on, welcome my dear friends. Now next 4 minutes, in 4 minutes this topic will be done. चलिए सबसे पहले हम स्टार्ट करते हैं. व्हाट इज द मीनिंग ऑफ इनिशिएशन इन दिस प्रोसेस? व्हाट इज द मीनिंग ऑफ इनिशिएशन? माय डियर फ्रेंड, जस्ट कंफर्म मी. हेलो एवरीवन, जस्ट कंफर्म मी. व्हाट इज द नेम ऑफ द एंजाइम डूइंग ट्रांसक्रिप्शन? व्हाट इज द नेम ऑफ द एंजाइम डूइंग ट्रांसक्रिप्शन? Very good, very good. Thank God, thank God. All is well, all is well. You people are with me. Oh my God, I'm impressed. Chalo, RNA polymerase. The enzyme doing the process here is RNA polymerase. Now listen here, one word. Hello. That enzyme RNA polymerase. Okay. Thank you Sweta. Thank you Pranaja. Thank you Pranshu. Now that enzyme which is doing the transcription process is RNA polymerase, and that RNA polymerase enzyme will come and it will get attached to this DNA पर आकर वो enzyme attach होता है. That is the reason I told you topics will become complicated. Now naming will become complicated. RNA polymerase attaching on DNA. Understanding? So we are having our DNA here, double stranded DNA, last and final time. Okay, आखिरी time we are having double stranded DNA. यहाँ पर enzyme आकर attach होता है और उस अटैचमेंट को ही हम इनिशिएशन कहते हैं हिंदी में. RNA polymerase enzyme will come and attach here on the DNA, and that itself is nothing but initiation in English. Okay, RNA polymerase enzyme ఇక్కడ వచ్చి అటాచ్ అవుతుంది DNA మీద, అదే ఇనిషియేషన్ అంటారు. సరేనా? సో నౌ లెట్స్ గో అహెడ్. వేర్ ఇట్ విల్ బి అటాచ్? ये कहाँ पर अटैच होता है? Now where is that enzyme attached? Remember, the enzyme will be attached before this sequence. I am writing here capital R. RNA polymerase enzyme will attach before the sequence, before this sequence. And wherever it is getting attached, that region is known as promoter region. And this promoter region, what you have to do right now is that, now it's your turn, you need to memorize something. So they are like 2 promoter regions or 2 regions in prokaryotes, and there will be 2 regions in eukaryotes. अब वो आपको मेमोराइज करना पड़ेगा. दैट वन यू नीड टू जस्ट मेमोराइज. आई डोंट हैव एनीथिंग टू डू इन दैट. कॉन्सेप्ट हो गया. आई रिपीट अगेन, व्हाट इज द मीनिंग ऑफ इनिशिएशन? इनिशिएशन मीन्स अटैचमेंट ऑफ दिस एंजाइम ऑन DNA. Oh my God. In prokaryotes, it will be attached at 2 places. So keep on writing here, sir. It will be attached at -10 sequence. Sir, what is the meaning of -10 sequence? Minus means before, 10 means 10 nucleotides before. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 nucleotides before. It will be attached 10 nucleotides before minus 10 sequence. And there the sequence will be thymine, adenine, thymine, adenine, adenine, thymine. So that enzyme will come and will search for that sequence thymine, adenine, thymine, adenine, adenine, thymine, and after searching for the sequence it will attach there. Okay, so that's the reason why doctors kept the name of minus 10 sequence as Tata box, or else this is also known as Prib now box. इसलिए मैंने क्या कहा था आपको, that's what I told you कि you need to memorize now. Minus 10 sequence is Tata box or Prib now box. And what is the sequence there? Thymine, adenine, thymine, adenine, adenine, thymine. Lovely. And I told you in prokaryotes there are two sequences. There will be one more that is known as minus 35 sequence. And minus 35 sequence here, minus 35 sequence here, yes, there the sequence will be T, T, G, A, C, A. That's all. Thymine, thymine, guanine, adenine, cytosine, adenine. That's also. Is it okay? And there is no other name for the minus 35 sequence. Thank God. And as I promised you, there are two sites for attachment in prokaryotes. Similarly, there are two sites of attachment in eukaryotes. So that you continue there only, sir. Below only you continue. I don't have place on this page here right now. So I will continue on the next slide. So in eukaryotes, we'll be having like two places of attachment again. Minus 25 sequence. Minus 25 sequence. And that minus 25 sequence is known as Hogness box. And it is same like your Tata box only, sir. And there is one more, that is minus 70 to minus 80. And minus 70 to minus 80 sequence, remember, it is also known as cat box. Why it is known as like cat box, sir? Why? Because there the sequence will be cytosine, adenine, adenine, thymine. Cytosine, adenine, adenine, thymine. So in eukaryotes, we'll be having minus 25 sequence. Okay, and minus 70 to minus 8. I hope you're understanding every time it is the same thing only. Minus means like before 25 nucleotides, before 70 nucleotides, something like that. I am done with the first step. Initiation हो गया. So I'll tell you for the last and final time. Initiation का मतलब क्या होता है? What is the meaning of initiation, sir? Enzyme is coming and attaching, and you have to memorize okay, where it will attach. In eukaryotes, minus 25, minus 70, minus 80, and in prokaryotes, minus 10 and minus 35. Okay, are you done with this one? Come on, please tell me, are you done with writing with this one? Yes. If you are done with the writing of this one here right now, guys, chalo, वापस आ जाओ. Welcome back here right now. I am done with initiation. Now welcome to elongation right now, sir. अब elongation ना sir, neither I have to teach you nor you people have to learn, sir. Elongation आपको पढ़ने का ज़रूरत ही नहीं है. Why? Why you don't have to learn elongation? I will tell you the reason. Look here. This enzyme will now start moving forward. After attachment, the enzyme will start moving forward. And after that, it is going to read. Again, the same thing. Reading will be in which direction? 3 dash to 5 dash. So point number 1, reading will be 3 dash to 5 dash. After reading, it is going to form RNA. Don't say new strand. New strand was in previous topic, replication. Now it is going to form from DNA, RNA. What are we learning? Transcription. Don't forget the topic. Chalo sir, now help me. Help me. It is going to form RNA, single strand RNA. मान लो this red colored one is RNA. Come on, help me. If it is gone in, cytosine. Cytosine, gone in. Cytosine, gone in. Gone in, cytosine. Thymine, adenine. Adenine, thymine. Hello. Will there be thymine in RNA? No. In RNA, the only one thing that you have to take care, wherever thymine, there will be uracil. Oh, that is the only one thing to take care. And again this is also formed from where only guys, from 5 dash to 3 dash. कितना आसान है ना. So reading will be 3 to 5 only. Formation of RNA will be like 5 dash to 3 dash only. Okay, the only thing that you have to be taking care about is wherever you are getting thymine, please replace that thymine with uracil. Please replace that thymine with uracil. That's the only one thing that we taken care, and that we have been learning from our school days. And in this way RNA will be formed, sir. RNA formation हो गया. Are you all done? That's why I told you elongation is almost the same copy paste, sir. Neither you have to study nor I have to teach you people. Now welcome to termination. Termination will be by actually two methods. One will be the row dependent. I hope you know this symbol, row. One will be row dependent termination. Another one will be like row independent termination. One will be row dependent termination, another one will be like row independent termination. Termination दो तरीके का होता है. So there will be two types of termination, row dependent and row independent. Now what is the meaning of row dependent, row independent termination? Sir, must time pass, sir, full time pass. Unnecessarily students will think molecular biology is very difficult. I think molecular biology is actually the easiest one, sir. Easier than our glycolysis Krebs cycle. वहाँ पर तो एंजाइम सीखना पड़ता है. यहाँ देखो स्टोरी, कहानी. It seems there is a protein here. The name of the protein is rho protein. Doctors kept the name of the protein itself as what, sir? Rho protein. And it seems that what is the function of rho protein? Rho protein का काम क्या है? Two function. Stop the formation of RNA. In some good English, terminate the formation of RNA. And after stopping or terminating the formation of RNA, it is also going to remove the RNA from here. Acha, sir. Okay. So that you know the two black colored lines are DNA. And in the middle that red colored line is RNA. So from there it is going to remove the RNA outside. Achha. So stopping the formation of RNA, removal of RNA is done by rho protein. That is why rho dependent termination. So rho dependent termination is done by rho protein, and rho protein is actually helping in two things. It stops the formation of RNA and removes the RNA. Stops the formation of RNA and removes the RNA. Andukosam, that's the reason why it is known as row dependent termination. Ante అయిపోయింది. सबको समझ में आ रहा है सर? Are you all done? Are you all done? Very good. Don't have to worry. So row dependent termination. Row dependent termination means yes, there will be a row protein, and row protein will help in stopping the formation of RNA and removing the RNA. That is why row dependent termination, sir. And now what is the meaning of row independent termination? Row independent termination means very simple. You already know that. Yes, I have to remove the row proteins. That means there is no row protein. No row protein, no row protein. That means what's a row independent termination? Row independent termination. Okay. And then sir, how will RNA stop the formation then? RNA itself is when there is no row protein, how it is stopping? RNA itself is going to form like a hairpin like bend. Okay. So therefore, if there is no row protein, it forms RNA forms hairpin like bend. So, ठीक है ना? RNA forms hairpin like bend. Okay. So row dependent termination, row independent termination. It's all like I'm telling you, na sir. It's all like must time pass. Okay, na must time pass. Kahani sir. Okay, बच्चों. Is it okay? So that's all about the transcription. And once you are done with this one, sir, the initiation, okay, and the initiation means what? Attachment of enzyme, and then elongation, and then termination. Once you are done with that, you can clearly see RNA is formed here, sir. RNA formation हो चुका है. Is it okay? No titan, nothing like that. चलो. Okay जी, हो गया. And that completes our second topic. That completes our, first topic here in protein synthesis. That is our transcription wala topic ho gaya, sir. I'm done with transcription. So tomorrow, any point of time, if you want to just revise the entire transcription process, some students will be thinking, pata nahi transcription kya hoga? What is transcription? Nothing. My students, from today onwards, easy. Just open this page, one page, initiation, elongation, termination. Initiation means attachment of enzyme, 2 places. Elongation, no need to learn, take it easy. Termination by 2 methods, row dependent, row independent. हो गया sir. That completes your transcription process. Transcription process class. Done, done, done. I'll give you 1 minute of time. Please revise that. Then we'll go to the next topic, sir. Ajay, I told you, Ajay, everything will be done by in another 1 hour, and then we'll take a complete break. Don't worry about that. By forming hairpin bend, there will be there's actually something more in depth in that one. That is, there will be a sequence known as palindromic sequence or not. That is not required now. Jayashri is asking, why don't you teach the rest of the Hare Bapre? I'm already leaving teaching in so many places, and you're asking me to teach other subjects also. Will there be again anatomy, sir? I don't know, sir. I did not schedule yet. Again anatomy hoga कि नहीं होगा? Will you teach the structure and organization? Nothing, not required that. Srinivas, not required that. RNA, same thing like that only single stranded. Chalo, sir. A lot of students want anatomy. Acha. Chalo, sir. If you are done with this one here right now, and now if you are done with this one here right now, now let us go to the second one here right now, sir. The post-transcriptional modification right now, guys. Let's go to the second topic right now. That is a post-transcriptional modification. Okay. Now, just now from the DNA, now, okay. किसी को clearly anatomy and biochemistry, it was posted in every group on my Facebook and everything. सर जी लगा लो फिर से क्लास एनाटॉमी की. अरे यार, I'm actually hurt. You people are like recording these that and all, so अच्छा नहीं लग रहा. I'm not liking it, so I'm not going to do it again. Yeah, I was actually sincerely I was actually sincerely thinking of helping students. Many students will take the help of this one and all, and you people are all doing all mischief things. I don't know. Chalo sir. Please don't do sir. Now once you are done with the transcription right now guys, let's get started. Let's begin with the post transcriptional modification sir. यहाँ तक clear हो गया? Come on sir, don't disturb yourself. यहाँ तक clear हो गया? Everything clear till here? Come on. Come on sir, this is the important topic sir. Please listen here carefully right now. Okay. Now what is going to happen right now guys, the post transcriptional modification, everyone listen here sir. 2 minutes it will be done. Please write on the heading there as the post transcriptional modification. The post transcriptional modification. Friends, Tanu is asking at what time class will be over today. Yeah, today it is only 8 o'clock right now. If you sincerely listen to me, I think the class will be done by 9 o'clock or 9:30, maximum 9:30. Okay, so another 1.5 hour class will be done and it will be done. Okay, so please listen here right now. Now post transcriptional modification, we are already having an RNA here right now guys. Just now RNA is formed here, and this RNA will be from 5' to 3'. We all know that RNA will be like single stranded RNA here right now guys, 5' to 3', and this RNA which is just now formed here is known as hnRNA. Already I told you in the overview. तो चलिए सर इसको मॉडिफाई करते हैं. Let us modify this one. Please write down there, extremely important topic, and you extremely important topic. And my dear friends, you can always expect one question from here sir. So hnRNA will be from 5' to 3' sir. Okay, one of the modification will be taking place at 5'. 5' is like head. On the head you'll be wearing cap, इसीलिए उसको कहते हैं 5' capping. That is the reason why it is known as 5' capping. And in 5' capping, the substance that will be attached here will be the 7-methyl guanosine. In 5' capping, what is the substance attached sir? 7-methyl guanosine. 7-methyl guanosine sir. ठीक है. So 5' capping means there is a substance attached, 7-methyl guanosine. हो गया sir, one modification done. I told you now it is like just 2 minutes topic. And 3' ends like a tail. Tail पर यहाँ पर ना tail लगा दो. Attach one tail here right now sir. So there will be a long tail of adenine, like many adenines attached there sir. So because there are many adenine, many means poly. Adenine starts with A, that is poly A tail. ठीक है. So at the 5' end there will be 5' capping, and at the 3' end there will be a poly A tail. At the 5' end there will be 5' capping, and at the 3' end there will be a poly A tail. Poly A tail. ठीक है. And the last and final modification that you have to learn in the middle. In the middle you'll be having some small parts here right now sir, and those parts are actually known as introns. And then the part in the middle here will be the exons. So I write here I for intron and E for exons. And remember, introns will be like non-functional, and whereas exons will be like functional. So therefore बोलो बच्चों क्या होगा? Which one has to be removed off? Which one has to be removed off? Intron. So therefore my dear friends, introns to be removed. Introns remove. And after removing the introns, ligate the exons, attach the exon. And that third modification is known as splicing. इसको हम कहते हैं splicing. Splicing means what? Splicing means removal of intron and ligation of exon. removal of intron and ligation of exon. Splicing means what, guys? Removal of intron and the ligation of exon, and that is splicing. And important thing is it is done by an enzymes, and those enzymes are actually known as spliceosomes. And spliceosomes, they're also known as snRNPs. And snRNPs, if you read it like multiple number of times, it will be like snRPs, snRNPs, 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 snRPs, like that. Okay, na? So spliceosome or snRNP or snRPs, these are the enzyme which is going to do what, sir? Splicing. Is it okay? Ho gaya. Modification number 1, 5' capping. Modification number 2, poly A tail. And modification number 3, splicing. After doing this, finally whatever RNA is formed here, that will be the fully modified mRNA. That will be the fully modified mRNA. If you are done, please let me know, sir. Okay, are you done? Chalo, sir. Now once you are done with this one, guys, the modification is done. And now, my dear friends, see, we'll do one thing here. Hello everyone here. Yeah, intron is not useful, it is non-functional. We'll remove the intron, sir. So now we are done with the transcription and we are done with the post-transcriptional modification here right now, my dear friends. And now, my dear friends, what we have to do is we need to study about translation. But I'm saying from last 5-6 years, guys, not at all a single question is coming from the translation process, sir. Okay, na? So I'll just tell you whatever are important topics from here onwards to be learned. Don't worry about translation, guys. Please don't waste your time in unnecessary things. I always advise. There is so much content available, so much subject available. Important thing is what to be learned, what to be left for the exam. Okay, so don't worry about translation. I'll teach you some important topics which are required here right now. So, okay, so please observe here right now, guys. Everyone look here. The first important topic that you have to learn is about the genetic code. Genetic code. First of all, what is another name of the genetic code? Genetic code is also known as codon. Genetic code is also known as codon. Codon. Now, what is the definition of the genetic code of the codon, my dear friends? Yes, right down there. It is a combination. It is a combination of 3 nucleotides which codes for an amino acid. It is the combination of like 3 nucleotides which code for an amino acid. Now, what is the meaning of this one here right now? I'll give you an example, you'll understand everything. I think you have been learning about this one, guys. AUG. What is AUG, guys? Yes, adenine uracil guanine. Yes, it is a combination of 3 nucleotides. Okay, it is a combination of 3 nucleotides. Okay, it's a codon, and AUG will code for an amino acid, and that is methionine. Yesterday we have learned like 20 amino acid. Out of the 20 amino acid, methionine sulfur containing AUG will code for methionine. Are you getting my point here right now? Genetic code or codon will be coding for an amino acid. It is a combination of 3 nucleotides. Perfect, done. We understood the definition. And now, my dear friends, for example, if this is the RNA here, right? If this one is your mRNA, messenger RNA, okay, from 5' to 3'. Okay, remember on this we'll be having like many codons. And remember, always the very first codon will be AUG. So therefore, this AUG is also known as the initiation codon. Initiation codon, or else this is also known as the start codon. AUG is also known as initiation codon. And remember, at the end of this, always will be having the either UAA or UAG or UGA. Okay, na? So UAA or UAG or UGA, these 3 are together known as your the stop codons, or in some good English they are also known as the termination codon. And because they are not coding for any amino acid, they are also known as the nonsense codons, guys. Nonsense codon. So remember, in an RNA, mRNA, always the initiation codon will be AUG and the termination codon will be your, yes, UAA, UAG and UGA. Okay, okay, are you done with that one here right now? And now my dear friends, this is what they will ask you. इसलिए मैंने कहा ना आपको, okay, don't waste your time in unnecessary topics. Whatever is they're asking in the exam, just learn that, more than enough. ठीक है? And now this codon has got some special properties right now, guys. 4 properties. Okay, so let us study those 4 characteristic properties. Heading लिखो, characteristic properties. That's something important nowadays, guys. Remember, number 1, the characteristic property is a codon will be universal. Okay, what is the meaning of universal, guys? A same codon, okay, codes for the same amino acid in all species. Okay, a same codon codes for the same amino acid in all species. For example, I gave you the example as AUG here right now, guys. AUG codon will be coding for which amino acid? It will be coding for methionine amino acid. Now that AUG will code for methionine in human beings like you and me. AUG will code for methionine in animals. AUG will code for methionine in dogs. AUG will code for methionine in plants. In every organism, AUG, the same codon, codes for same amino acid in all species. Okay, that is the reason why a codon is universal. Number 2, whenever you are writing some codon, for example AUG, UAA and UGA, something like that, whatever codons you are writing, you are not allowed to use any punctuation marks in the middle. Some students are like, acha, sir said it is a combination of 3, after AUG comma, after UA full stop. No, you are not allowed to use the punctuation mark. So therefore, comma less, it will be comma less. And not only comma less, acha, sir asked not to use comma in the middle. Chalo, let us overlap that. You cannot even overlap. It will be non-overlapping. Non-overlapping. So therefore, you cannot use any punctuation marks in the middle, sir. No commas and all. And so therefore, non-overlapping. Okay. And number 3, remember the codon will be specific, guys. Specificity. And this specificity is also known as unambiguous. Now what is the meaning of that? Yes, AUG will code for which amino acid? Methionine. So remember, it is specific for methionine only. That's all. It's not like yesterday AUG is coding for methionine. Today morning it is coding for glycine. By evening it will code for lysine. ऐसा नहीं होता है. Amino acid will not change. It is specific to particular amino acid. And number 4 property, a codon is degenerate. And how to understand this one with number? My dear friends, in total how many codons do we have? We are having like 64 codons. How many codons do you have, guys? 64 codons. But in total how many amino acids do you have? You have only 20 amino acids. Come on, guys, ulta dekhiye. One amino acid can be, yes, one amino acid can be coded by more than one codon. There in total how many codons? 64 codons. So therefore less amino acids. So therefore I can say that one amino acid can be coded by more than one codon. Codons zyada hai. We are having like more codons. That is why that property is known as degenerate. One amino acid can be coded by more than one codon. That is known as degenerate. So my dear friends, nowadays trending questions you have to remember. Yes, a codon is universal, commaless, non-overlapping, specific, unambiguous, and degenerate. That is what they are asking in the exam nowadays. Is it okay? Are you all done? Come on. So this is one topic which is important for us, guys. Okay, codon wala topic. After this, which topic is important, guys? Please write on the heading there as RNA. RNA. We have got like different type of RNA. Messenger RNA, ribosomal RNA, transfer RNA, and all those different types of RNA. Out of that, just remember these points, sir. Can you please tell me, out of all those RNA, which one is the most abundant RNA? Which one is the most abundant RNA? Most abundant. It is your Ribosomal RNA. Ribosomal RNA is the most abundant RNA. Okay, na? Because every cell will have ribosome and ribosomes are like abundant ones. Okay, na? And which one is the smallest RNA? That was one more important question. Smallest RNA, that will be your tRNA, sir, transfer RNA. Okay, and out of all the RNA, my dear friends, don't remember like each and every RNA, just remember about the transfer RNA, sir. So please write down the cell heading as tRNA. Okay, let us learn about the structure of tRNA. Do you know what is the shape of tRNA? The shape of tRNA will be like clover leaf shaped. Shape of tRNA will be like clover leaf shaped. It will be clover leaf shaped. Clover leaf shaped. Okay. So therefore, please draw the diagram of the tRNA, guys. That's important. And in details to be remembered, this is 5' end and this will be 3' end. Okay. At 3' end, you have to remember the sequence. The sequence will be, yes. Anyone, any idea? Come on. It will be cytosine, cytosine, adenine. Cytosine, cytosine, adenine. That is the question number 1. Chal. Question number 2. At the 3' end, it is going to attach amino acid. Amino acid will be attached here. So because it is accepting amino acid at 3' end, that arm is known as acceptor arm. इस arm को हम कहते हैं acceptor arm. At 3' end, it is going to accept the amino acid, therefore acceptor arm. Exactly opposite arm is for reading the codon. Therefore, this arm is known as the anticodon arm. Anticodon arm. So 3' end will be acceptor arm and the opposite arm will be the anticodon arm. And here, this part here right now, one more arm here right now, actually that is not needed. To be very frank, for your MCA exam, if you learn this acceptor arm and if you learn anticodon arm, more than enough, sir. More than enough. Now, for some students who are like very curious, yes, this arm here right now, will be dihydrouridine arm. Just remember DHU arm, and this will be like T. You remember symbol psi? T psi C arm. And if you are still unsatisfied, not satisfied, sir, I want more. Yes, sometimes present, sometimes absent. There will be one more arm, and that will be variable arm. कभी-कभी होता है, कभी-कभी नहीं होता है. Enjoy. But remember, my dear friends, if you are targeting your MCA exam, remember 3' and acceptor arm, and exactly opposite of that anticodon arm. So that is more than enough, sir. Okay, that's all. So in RNA, if you remember this much information, more than enough, sir. So it's like, almost we are at the end right now. We are having like small topics. So codon completed, RNA completed. Shall we go ahead right now? Are you ready? Are you understanding, guys? Tell me yes or no. Anticodon arm means that is going to go and read the codon. वो जाकर codon को read करेगा. हो गया बच्चों? चलो यहाँ देखो सर. Now once you are done with this one here right now, my dear friends, चलो सर इसको खत्म करते हैं. This topic will finish it off. I told you very clearly. I told you learn transcription in detail, learn post translation modification in detail. I told you very aram se slowly. Don't worry about translation right now, guys. Okay. From last 5-6 years, there is no question at all. They're not asking any question from there. Don't worry about that. Just now worry about this topic, that is the post translational modifications. ये पढ़ते हैं और इस topic को खत्म करते हैं. Let us finish this topic. So all like small topics. So please write down the heading there as the post translational modification. What is your doubt, brother? Bharat, I told you very clearly, CCA, cytosine cytosine adenine, is at 3' end, and whatever initiation you are telling me, initiation will begin from AUG only, and AUG will be at 5' end, not 3'. And second point, that will be mRNA, messenger RNA, not tRNA. Concentrate. Okay, welcome here right now, sir. Now welcome to the post translational modification. Subhash, अरे यार तुम लोग सुनते क्यों नहीं हो? I told you very clearly, don't waste your time in learning translation right now. They are not asking any questions in that. Don't learn the topic. Don't study the topic. Now, sir, for the last and final time, DNA is getting converted. Yes, DNA is forming your hnRNA, heteronuclear RNA. Heteronuclear RNA is modified towards a mRNA. Now this mRNA will come out of the nucleus into cytoplasm. mRNA is forming your protein. Okay, and now I am speaking about this protein modification, post-translational modification, important topic. You imagine one of the protein, that is collagen protein in our body, and you have to learn the modification of only the collagen protein, sir. See, now here also some students will be like questioning. Actually ना पंगा ये है कि in our body everything is like almost protein only. Just enzymes nature, proteins. What is the nature of hormones? Protein. Transporter, protein. Receptor, protein. And ion channel, protein. Muscles, protein. Bone is made up of collagen protein. So if you just observe in our body, everything is like protein only. The nature of almost everything is like almost protein only. So therefore, my dear friends, is it possible to learn the modification of every protein? Is it possible to learn the modification of each and every protein? Not at all possible. So therefore, you have to learn for the exam the modification of collagen protein. Which protein? Collagen. And now please write down there, yes, the post-translational modification of collagen. Likho, sir. What is the modification in collagen? Hydroxylation. And where is hydroxylation taking place? At lysine and this khatarnak English. Look here, my dear friends, what is collagen? It's a protein. Protein means what? Polymer of amino acid, made up of many amino acid, and somewhere in the middle you are having an amino acid known as lysine. Again it is made up of amino acid. Somewhere in the middle you are having proline, something like that. Understanding? So this is like our imaginary diagram, sir. So collagen is made up of like many protein, many amino acid. I repeat again, collagen is a protein, and we have already learned yesterday in detail what is a protein made up of? A polymer of amino acid. And somewhere in the middle we are having which amino acid? Lysine. And somewhere in the middle we are having proline. And on this, what is happening on lysine and proline, guys? Hydroxylation. And my students are expert. What is the meaning of hydroxylation, guys? Addition of OH group. So OH group is added here. OH group is added here. Oh. Now read the statement. Hydroxylation at lysine and proline. You people know very well what is the name of the enzyme. Hydroxylase. Complete name, lysyl. What is the name of the enzyme here also, sir? Hydroxylase. Complete name, prolyl. So which enzyme is helping in addition of OH group? Yes, it is hydroxylase enzyme. Lysyl hydroxylase, prolyl hydroxylase. And remember, these two enzymes will be requiring coenzyme, and that coenzyme is vitamin C. And that is vitamin C. Oh my God. Please turn back the pages and see yesterday's class. I taught you about vitamins topic, and in vitamin I taught you the last one, vitamin C. देखो turn करके देख, turn the pages and see vitamin C first function हमने क्या लिखा था? The first function that we wrote there yesterday was, yes, it is helping in post-translational modification of collagen. Can you see vitamin C helping here? Yes. Now after making a chain like this, not only one, you have to make like 3 chains like this. And after making 3 chains like this, first do addition of OH, addition of OH, addition of OH, wherever lysine proline add kar do. And now what you have to do, these 3 chains you have to make them helical. One chain here, second chain here, and the third chain here, and that is known as the triple helix. And triple helix, yes, it is done by which enzyme? That is important, sir. And that will be your lysyl oxidase. And this enzyme recently asked in MC exam, it requires copper. This enzyme requires what, sir? Copper. Copper चाहिए उसके लिए. ठीक है जी. तो कोलाजन पहले एक चेन बनाओ, फिर उस चेन के ऊपर OH ग्रुप ऐड करो, एंड आफ्टर एडिंग द OH ग्रुप्स इन दैट मेक अ ट्रिपल हेलिक्स ऑफ दैट, एंड ट्रिपल हेलिक्स इज फॉर्म्ड बाय लाइसिल ऑक्सीडेज. और उसके लिए क्या चाहिए सर? कॉपर. Now your collagen is completely ready to be utilized in body. So in our body the most common protein collagen, इसीलिए only you have to remember the modification of collagens. Are you done with that? Are you all done with that? So these are all like, you know, small topics that you need to remember, guys. One is about the codon, another one is about the RNA, and another one is about the modification of collagen. बच्चों come on, हो गया? Are you done? Please confirm me. चलो sir. If this is done here right now, okay, one single point here right now, sir. DNA to RNA and mRNA to protein, and after the formation of this protein, remember these proteins will be folded, sir. Protein folding. Can anyone tell me the protein folding is done by which substance? Yes, the protein folding is done by, it's all like, you know, miscellaneous points. Protein folding is done by which one? Yes, the substances which are known as try to pronounce that. I don't know why many students will be saying, you know, chaperon करते रहते. It's not chaperon actually, it is chaperons, you know, chaperons. Yeah, it's not chaperon, it's chaperons. Okay, खतरनाक लोग होते हैं सर इंग्लिश में तो. ओ माई गॉड. ओके, रिसेंटली आई वाज एट वन ऑफ द एयरपोर्ट, मैं कहीं पर था, एंड आई वेंट टू दैट, यू नो, वो होता ना KFC खाने गया था. आई थिंक सम पीपल आर हंग्री राइट नाउ, और भी मज़ा आएगा. So I went for eating KFC and all those, and in that, you know, one fellow came there and he was like, I want Zinger box, and in Zinger box don't give me Pepsi Cola, give me Virgin Mojito. Had they too much Virgin Mojito? It's Mojito, it's not Mojito. It is written as J there, but you have to spell as H. And similarly this one, sir, chaperon not chaperon, it's chaperons. Okay, so chaperons are helping in what, sir? Protein folding में help करते हैं sir. Okay, and now the last and final thing, sir, blotting techniques. सर किर्गिस्तान कब आओगे? सर आप बुला लो, मैं अभी आ जाऊंगा भाई. There will be a northern blotting technique, okay, and then there will be a southern blotting technique, so there will be a northern blotting, and then there will be a southern blotting and western blotting techniques. Okay, and you just have to remember this blotting technique is useful for what is the function of this one? Northern blotting technique, remember, it is useful for, you know, analysis of RNA. Southern blotting technique is useful for DNA, and western blotting technique is useful for proteins. So that is the only thing that you have to remember in this blotting technique, sir, nothing else. Okay, so which blotting technique is used for what? Northern blotting is used for RNA, southern blotting is for DNA, and western blotting is used for the proteins. But how to remember without confusion for long term? Remember, in North India people love to eat roti like me. In South India people love to eat dosa like you. And in Western countries the people love to eat neither roti neither dosa, pizza. Is it okay? So therefore remember, Northern blotting it's for RNA, southern blotting will be for DNA, and then western blotting technique is used for the, yes, the protein cell. That's all. So if you remember this much information, that will be done about about the molecular biology. Molecular biology हो गया इसका. Is it okay? Done? And after this, my dear friends, we need to learn about the topic of enzyme. So enzyme topic will take another 20-30 minutes. It will be done, sir. So shall we complete that or shall we take a small break? Sir, small break. Break. अच्छा चलो ठीक है sir. We'll take a break exactly of 15 minutes, sir. Okay? It's 8:30 right now. We'll start at 8:45. 8:30 right now, sir. We'll start from 8:45. ठीक है? In enzymes topic, guys, first of all, what is the definition of enzyme here right now? We all know that enzyme is a substance which is going to increase the velocity of reaction. Enzymes are those which are going to increase the velocity of the reaction, guys. So enzymes are going to increase the rate of the reaction. Okay, enzymes are going to increase the velocity of the reaction. So these kind of words will be used for, or the speed of the reaction. तो जो velocity या फिर speed और rate of the reaction बढ़ाएगा, जो increase करेगा, ठीक है? So that substance is known as the enzymes. So enzymes are those substances which are going to increase the velocity of the reaction. Done? Now the next thing that you need to learn after definition is nature of enzyme, guys. So please write on the heading there as nature of enzyme. Now what is the nature of enzyme? We all know enzymes will be protein in nature. What is an enzyme made up of? Protein. But along with the protein part, there will be sometime even the non-protein part also made up of this. And remember, yes, then we need to learn okay, what is the name of the protein then, sir? Remember, the protein part of the enzyme is known as apoenzyme. The protein part of the enzyme is known as apoenzyme, and the non-protein part of the enzyme, it will be known as coenzyme. Okay. The protein part of the enzyme is known as apoenzyme, and non-protein part of the enzyme is known as coenzyme. And remember the another name of this one, the coenzyme is known as prosthetic group. The coenzyme, it is also known as the prosthetic group. Or else it is also known as the cofactor. So whatever coenzyme I'm telling you right from the beginning of the biochemistry, guys, what is the meaning of coenzyme? Coenzyme means it is the non-protein part of the enzymes. So normally proteins, enzymes are protein in nature. That protein part is known as apoenzyme, and the non-protein part is known as the coenzyme. And remember, these two together, apoenzyme and coenzyme, protein part and non-protein part together, it will be known as the holoenzyme. It is known as the complete enzyme or the whole enzyme or holoenzyme. So the protein part will be known as the apoenzyme and the non-protein part is known as the coenzyme, guys. And together it is known as holoenzyme. And that was asked in the MC exam. Take care about that. Okay? And that completes the definition, that completes the nature of enzyme. And now welcome to the most important thing, that is the factors affecting the enzymes. Number one factor that will be affecting the enzyme will be concentration of enzyme, and number two will be the concentration of substrate. Number three will be concentration of product, and number four will be the temperature, and number five will be the pH. These are the five factors we need to learn which are affecting the velocity of the reaction. And now my dear friends, please help me right now. You have to help me right now, guys. For example, there is some reaction, substrate is getting converted to product in the presence of enzyme. In general, some reaction is going on. There is a substrate getting converted to product in the presence of enzyme. Now for example, what is the meaning of concentration, guys? Concentration means what? Amount. In simple English, concentration means what's the amount. If concentration of enzyme increases, अगर मान लो कि amount of enzyme बढ़ता है, if concentration of the enzyme increases, amount of enzyme increases, concentration of the enzyme or the amount of enzyme increases. Now tell me what will happen to velocity. Velocity बढ़ेगा कि कम होगा? Velocity of the reaction will be increasing or decreasing? Yes. From the definition itself we'll know that what is an enzyme. Enzyme is a substance which is going to increase the velocity of the reaction. अगर आप enzyme बढ़ाओगे, if you increase more enzyme, then what will happen? More will be the velocity. So therefore it is directly proportional to velocity of reaction, or simply you can write as capital V. So more the concentration of enzyme, more will be the velocity of reaction. Similarly, if you increase the concentration of substrate, then what is going to happen? More the, अगर मान लो if I'm having 10 substrate molecule, out of that 10, 8 are converting to product. If I increase that to 100 molecules, okay, and out of that 180 molecules are getting converted to product, okay, that means that more the substrate and more will be the velocity of the reaction cell. So therefore directly proportional to velocity. And now welcome to the last and final condition right now, guys. What about the product right now? अगर आप product बढ़ाओगे तो क्या होगा? If you increase the product, then what is going to happen? Velocity of the reaction will decrease. Why? Why? Because it's common sense. When I'm already having like more product, will I think of making or increasing more velocity of the reaction? No. Already I'm having a lot of product there. So therefore, more the product, less will be the velocity, guys. Inversely proportional. So that means the final conclusion, more the enzyme, more will be the velocity. More the substrate, more will be the velocity. But more the product, less will be the velocity of the reaction, guys. That's all. So this you have to remember. And now what about temperature? अगर हम temperature बढ़ाएंगे तो velocity क्या होगा? बढ़ेगा कि कम होगा? If you increase temperature, what will happen to velocity of the reaction? For doing that, let us just take a graph here right now, guys. On the x-axis, I'm going to take temperature, and on the y-axis, I'm going to take the velocity of reaction. I mean to say capital V. Now, my dear friends, what is going to happen here is that when you are increasing temperature, that means what is going to happen, sir? You are giving heat energy, and whenever you're giving energy to anything, it will become more activated and it will be increasing. So therefore, when you increase temperature initially, yes, velocity increases. But if you further increase temperature, you already know, yes, you already know the nature of enzyme. It is actually protein in nature. अगर आप और temperature बढ़ाओगे, the protein will undergo denaturation. And when the protein is denatured, that means enzyme is gone. When the enzyme itself is gone, will the velocity increase? Ulta, it will decrease. So therefore, my dear friends, with respect to temperature, what is going to happen? You know, initially velocity increases, reaches to maximum. If you further increase temperature, velocity decreases. So never say increase or decrease. Yes, always say that you'll be getting a bell-shaped graph. With respect to temperature, don't say it is increasing or decreasing. Don't say that. Always say you're going to get bell-shaped graph. That means first increases, reaches maximum, then decreases. Okay. And my dear friends, you can clearly see here on y-axis we have taken what's our velocity, and this is the place where you are getting the maximum velocity. So this one will be known as the Vmax, maximum velocity. That is the place where you are getting the maximum velocity. Okay. And with respect to pH, exactly copy paste. Exactly आपको ना क्या करना पड़ता है? Copy paste करना पड़ता है. You need to do the copy paste. So therefore, my dear friends, on the x-axis take the pH, and on y-axis you can take the velocity of reaction. Capital V. Okay. And now what is going to happen with respect to pH? You already know the pH scale. pH scale will be like, you know, that 1 to 14, you know, acidic, basic, neutral and all. तो जैसे आप pH बढ़ाओगे, what is going to happen? Initially velocity increases, then reaches to the maximum, then again it is decreasing. Again you are getting bell-shaped graph, and here is the maximum velocity, sir. Vmax. So this topic is like so simple, guys, here right now. Yes. Concentration of enzyme increases, velocity increases. Concentration of substrate increases, velocity increases. Concentration of product increases, velocity decreases. And with respect to temperature and pH, we are getting bell-shaped graph. First increases, maximum, then decreases. That is what you have to remember. Okay, done. And now let us work out in detail with the concentration of substrate here. Ye wala part. This is something important. Okay. Let's work in detail. Please write down the heading there as Michaelis-Menten graph. Michaelis-Menten graph. Michaelis-Menten graph will give you the relation between concentration of substrate and the velocity. And you already know concentration of substrate is directly proportional to velocity of the reaction. So if it is directly proportional to velocity, let us take a graph. On x-axis I am taking substrate concentration, on y-axis I am taking velocity of reaction. Is it okay? And we can clearly see there, yes, what is the relation between substrate and velocity, guys? Directly proportional. So therefore what is going to happen? As you increase substrate, velocity will also increase proportionately. Therefore you will be getting a parabolic graph like this, guys. Okay, it is increasing proportionately. And in this graph, where are you going to get the Vmax? Yes, somewhere here. This will be the maximum velocity. Wow, very simple, nothing to worry. Unnecessarily students will be getting feared with graphs and all. Nothing to worry about the graph, guys. Michaelis-Menten graph is giving you the relation between substrate and velocity. And remember, you will be getting a parabolic graph, directly proportional. And here, the most important thing here will be the constant. There is a constant which is known as the Michaelis-Menten constant. Michaelis-Menten constant. It is represented by Km. Michaelis-Menten constant is represented by Km. Now what is the definition for this Michaelis-Menten constant? Write down there. It is the concentration of substrate at half the maximum velocity. Michaelis-Menten constant Km is actually the concentration of the substrate at half the maximum velocity. Concentration of the substrate at half the maximum velocity. We'll do one thing here. Read that definition word by word in English and apply on the graph. Let's apply on the graph. What is the condition given? Half of the maximum velocity. Okay. Maximum velocity यहाँ है. Half of the maximum velocity somewhere in the middle here right now, sir. Okay, that is Vmax by 2. Half of the maximum. At that, it is the concentration of substrate only. Acha. So from here, welcome to the concentration of substrate. Okay. So this concentration of substrate itself is known as what? Km. Okay, that's all. It's so simple. Is it okay? So guys, make your life very simple here. What is Michaelis-Menten constant? It is nothing but substrate concentration only. It is nothing but concentration of substrate only. But when at half of the maximum velocity, that is the important condition. Is it okay? So, aapki nazar na, you people have to always have, in your mind these 4 things: concentration of substrate and Km on x-axis, whereas the velocity of the reaction and the Vmax will be on the y-axis. Clear? So Km is nothing but the concentration of the substrate at half the maximum velocity. Km is nothing but the concentration of substrate at half the maximum velocity. Okay, done. And now you might be thinking, sir, what is the use of this one? चलिए sir, मैं आपको बताता हूँ इसका use क्या होता है. What is the use of this one? Please write down the heading there as next topic, enzyme inhibition. Enzyme inhibition. There will be like 2 types of enzyme inhibition. Number 1, it will be like a reversible inhibition. Another one is irreversible inhibition. Enzyme inhibition ना 2 तरह का होता है. One will be reversible enzyme inhibition. Another one is irreversible enzyme inhibition, guys. And I want all of you to please, okay, I want all of you to, yes, please stop writing here right now, guys, and please observe here right now. Reversible and irreversible, okay, enzyme inhibition, no need to even write down, it's there in the name itself. I'm having substrate, it's getting converted to product in the presence of enzyme. Normally reaction is going on. I meant to say normally means what, sir? Normally means what? Without inhibitor. Abhi inhibitor nahi hai. Now we are going to introduce the 4th player here, that is inhibitor. Inhibitor is coming here, inhibiting the enzyme. enzyme inhibited and the reaction will be stopped. Okay, normal, okay, understood. Inhibitor will come there and inhibit the enzyme, therefore reaction stop. But after some time that inhibitor will withdraw from there, sir. The inhibitor will go away from there. If it is going away from there, again the enzyme will start. Which type of inhibition? Reversible. Understanding here? So inhibitor coming, inhibiting, and again it is going away from there, sir. So therefore which type of inhibition is this one? Reversible inhibition. No need to study anything else. Now second category, we are giving a very strong inhibitor, and it is such a strong inhibitor, it is going to inhibit this enzyme forever. It's not going away from here. That inhibitor is not withdrawing from there, so therefore that is irreversible inhibition. It is inhibited forever. So therefore my dear friends, enzyme inhibition दो तरह का होता है. It will be of two types: reversible, irreversible. You don't even have to worry about their definition. They will never ask you in the exam. It is there in the name itself. What they will ask you in the exam, and they will ask you pakka in the enzyme, yes, is welcome to reversibles. Reversible will be the competitive and non-competitive inhibition. So again it is divided into two categories. Okay, one will be the competitive inhibition, another one will be the non-competitive inhibition. Okay, sir. Now what is the meaning of this competitive and non-competitive inhibition? Yes, that inhibitor is actually competing with the substrate. Inhibitor is competing with the substrate. Now what you have to remember, just two golden words that you have to remember for competitive and non-competitive. Yes, remember, in competitive inhibition, my dear friends, Vmax will remain same. The Vmax value will not change. What do you mean by remaining same? Before adding inhibitor and after adding inhibitor. So before adding inhibitor we are calculating Vmax value. After adding inhibitor we are calculating Vmax value, and it is the same. But whereas in competitive inhibition what will happen? Vmax will remain same, Km value will increase. These two golden words you have to remember for competitive. And for non-competitive inhibition, remember exactly opposite. Km value will remain same before inhibitor, after inhibitor, whereas the Vmax value will decrease. Exactly opposite of that. So remember one thing, opposite will automatically come, sir. And I'm guaranteeing you in the coming June exam, pakka questions, definite question. You have to remember these two words, sir. In competitive inhibition, Vmax same, Km increases. And then in the non-competitive inhibition, yes, what will happen, sir? The Km remains the same and the Vmax decreases. Are you okay with that here? Examples, someone is asking me examples. Guys, examples you will study in pharmacology. Is it okay? In pharmacology, you will be giving drugs. For example, you are giving allopurinol. Allopurinol is a drug. So that drug, allopurinol, it's acting like your inhibitor. Allopurinol is a drug. It's acting like your inhibitor, and that is going to come and inhibit the enzyme. Which enzyme? Xanthine oxidase. And that is used for the treatment of what? That is used for the treatment of gout. Understanding here? So there they will tell you that allopurinol is acting like a competitive inhibitor, non-competitive inhibitor, and all those things. Okay, now. So now we cannot go in that examples of pharmacology. Otherwise, we have to tell you about the drug. Then it is used for which disease. That's totally a different story in pharmacology. So here in my biochemistry, learn the concept and apply the concept in your pharmacology next subject. Understanding, my dear friend? In biochemistry, pakka definite question in the June exam. Take my words. Competitive inhibition, they will ask you Vmax same, Km increases. Non-competitive inhibition, remember Km same, Vmax decrease. Vmax value will decrease. Is it okay? Come on. Are you done? Very good. Now welcome to the last and final topic in enzymes, guys. Please write in the heading as isoenzymes. Guys, what is the meaning of iso? Iso means same, not single. Iso means same. Okay, it will be the same enzyme. Very good. It will be the same enzyme, but in different tissues. I'll give you example. Listen here right now. Example. The best example for the isoenzyme is LDH. You know LDH? Lactate dehydrogenase. And lactate dehydrogenase has got 5 isoenzymes: LDH1, LDH2, LDH3, and then LDH4, and then LDH5. It is the same enzyme only, LDH only. It will do the same reaction, pyruvate to lactate, lactate to pyruvate. हमने already पढ़ा है. So it will be the same enzyme and it will be doing the same reaction, but it will be differing in the tissues. Which tissues? Remember, out of all this category, LDH1 and LDH2 will be more in your heart, whereas LDH4 and LDH5, this will be more in the skeletal muscle. Okay? And remember, LDH3, this will be more in your, like, present in brain, RBCs and all those things, kidney and all. Understanding here? Various organs. So what I am trying to tell you is that it will be the same enzyme doing same reaction. Okay, na? Same enzyme doing same reaction, but in different tissues. So LDH1 and 2 will be in the heart, and LDH4 and 5 in the skeletal muscles and all. No, Divya, you are giving a wrong example. I told you hexokinase, glucokinase are two separate enzymes. They are not like isoenzymes. No, not at all. Don't take it wrongly. So this is an example of, like isoenzymes and all. Now listen here right now, sir. 1 second. Now why this topic is important, I'll tell you here. It is important because of clinical conditions, sir. Okay, now. LDH is important, the lactate dehydrogenase. We have learned this in the carbohydrate metabolism, anaerobic glycolysis. Now this LDH enzyme is important. Come on, if you have learned pathology, myocardial infarction. Guys, in normal people like you and me, LDH1 will be more when compared to LDH2. In normal people like you and me, LDH1 will be more when compared to LDH2, sir. If you do a test right now in your body and my body, this will be done. Suppose a person got a heart attack, myocardial infarction. Yes, what will happen? Suddenly it will be flipping. In MI, in MI, what is going to happen, guys? LDH2 will be more, guys. LDH2 ज़्यादा होता है, ठीक है? So in MI, what is going to happen? LDH2 will be more. Are you getting my point here? And that is the reason why the moment someone is getting heart attack, you just do a blood test and you will see, अच्छा LDH2 ज़्यादा हो गया, declare, अच्छा इस patient को ना अभी recently heart attack हुआ है, something like that. So that will be useful for you in your pathology. Study that. But in biochemistry, what is my concept here right now, sir? You have to remember how many isoenzymes? 5 isoenzymes. Heart will have LDH1 and 2, and the skeletal muscles will be having LDH4 and 5. Perfect, done. Now last and final, one more example important is creatinine kinase, CK, and this will be having like 3 isoenzymes: creatinine kinase MM, and creatinine kinase BB, and the creatinine kinase MB. What is the meaning of that, sir? MM means it is found in the muscle, skeletal muscle, and BB, this will be found in the brain, and yes, again this MB, sir, creatinine kinase MB will be found in the heart. And फिर से वही, go into your second year, learn pathology. In pathology, myocardial infarction, जब heart attack होता है, तो this enzyme will be elevated in the blood. So all this will come in your clinical condition. So these two examples of isoenzymes are important. LDH has got the 5 isoenzymes, and then CK, that is creatinine kinase. Creatinine kinase has got only 3 isoenzymes: CKMM, CKBB, and CKMB, sir, in the skeletal muscle, in the brain, and the heart. Are you done with this one here? So these are all the topics that you need to learn about the enzyme topic completed, sir. Okay, so isoenzyme, and then enzyme inhibition, reversible, irreversible, competitive, non-competitive, and then you have to learn about the Michaelis-Menten graph and constant, and then you have to learn about all the factors affecting the enzyme activity, nature of the enzyme, proteins, that is apoenzyme and coenzyme, and then lastly definition. So this is done with the topics. It's a very easy topic. And guys, I have sent you the LMR. In the LMR, last minute revision, I have given you the examples of non-protein. Okay, you remember coenzymes? Already many coenzymes we have covered. Vitamin C is a coenzyme for lysyl hydroxylase and all, है ना? Biotin is a coenzyme for 3 enzymes. You remember? Vitamin B12 is a coenzyme. Remember? So my dear friends, that is nothing to teach over there. You have to remember which enzyme requires which coenzyme. For example, pyruvate carboxylase, it requires biotin. So you need to memorize that. So in that LMR last minute revision, at last I have given you a list of enzyme and the cofactor, enzyme and the coenzyme, enzyme, coenzyme, enzyme, coenzyme. There's a table made, sir. And not only that, in that LMR, if you see just in 3 pages, entire biochemistry covered, sir. Okay, now we are done with the rate limiting enzyme. Okay, yeah, of course, copper is required. So we are done with the rate limiting enzyme. In one table, I have given all rate limiting enzyme. In one table, I have given all the locations. In one table, I have given you all the important amount. So do one thing, take a print of that and please stick that wherever you are studying. आप जहाँ जहाँ पर भी पढ़ाई कर रहे हो ना, वहाँ पर आपके ना स्टिक कर लो वहाँ पर, ठीक है ना, एंड जस्ट कीप ऑन रिवाइजिंग दैट एंड यू विल नेवर 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 forget that one, sir. Okay, so please take a print out of that one. Only 3 pages, sir. Only 3, not even like 3 pages, only 3 sides. Okay, just 3 sides. You just stick wherever you are learning and keep on revising every day, sir. List of all the location, list of all the rate limiting enzyme, list of all the diseases, list of all the amino acid and important products and all those things. So I think that will be like easier for you. Okay guys, and so please guys, okay, whatever I taught you, please learn that. That will be more than enough. I'm telling you, I have seen like thousands and thousands of students in my life. Again the same thing, guys. They will listen, they will just listen to their friends and all. वो आकर बोलेगा अरे यार ये book में दिया है, that book is given that, this is book is given that, and again the same time after the exams it will be like sir, all the questions came from your notes. You have seen like many feedbacks on my Facebook. So please, every time the same thing, sir. There will be hundreds of messages coming to my WhatsApp. Sir, इस book में ये लिखा है, इस book में ये लिखा है. Please guys, whatever I told you, please learn that. That will be more than enough. Is it okay? So please don't get diverted and don't get distracted and don't think negatively कि यार something is missing. So please don't do that. So therefore, whatever I taught you, please learn it. Hope this session was useful to you people, guys. Anatomy and biochemistry part is done, and we are going to stop the class here. Each and every topic that is required has been completed, sir. Thank you, sir. And anytime if you have any doubts, sir, please do one thing, drop a message in the that WhatsApp group में ना message drop कर दो, and that will be more than enough, sir. Okay, so whatever I have given you the notes, and please don't message me. One thing, please don't message me the doubts individually, guys. I'm sorry if I'm not able to answer you because there are so many messages in the WhatsApp. I can't answer them. Just drop your message in the group. I'll surely come back to you. Okay, so please drop your message in the group only. Okay, thank you. Okay, thanks a lot, sir. I hope you people have enjoyed. Happy helping you all. I'm really happy that I've helped like so many students in different places. So please make use of these classes and try to revise everything, guys. Okay, and please don't misuse, guys. Okay, now please don't misuse. If you have recorded or something like that, please, I'm requesting you all, don't do that. It's very bad.